even the organism that causes infectious bacteria, virus, fungi, and protozoa, elements, these are the major, due to the environment is contaminated with water. So these are the sources, different means. For example, mosquito, or food, or water, major air. These are, they reaches the person, and you can see the bottom level. Arrow marks is irreversible, man to human, woman to embryo. Today the miscarriages and so much of gynecological related problems are much influenced in present generation due to the environmental toxins. As per the NIN, National Institute of Nutrition, Hyderabad, they are given a shockable information that after 15 years of research, in the areas of Hyderabad and Secunderabad, the miscarriages are very common. And gynecological problems are very high last 10-15 years. Why? What is happened? The Medical Council of India, they given the report that the people who are in and around that area, they're consuming more grapes. Grapes in the sense of the cultivation of grapes, etc. They use a huge quantity of 4 cycles of pesticide spray. That to 100% spray. So that's why most of the pesticides are not water soluble. So these people are consuming these aspects which enter in the gonadial development, gametogenesis process. It interferes with sex hormones, etc. That's why such type of chromosomal aberrations, very pathetic to say. Even when the same NIN said, that National Institute of Hyderabad says, the mother milk also having the residues of pesticides, heavy metals. Even the amniotic fluid, the mother is carrying the embryo growing in the fetus, where the fluid, allantochorion amniotic fluid, that fluid gives a cushioning movement for the mother while carrying the baby. The mother fluid, the baby which is growing in the womb, in the fetus, that fluid also containing the residues of pesticides and heavy metals. It is a shockable incident. Even recently in the Punjab farmers, their blood having the residues of heavy metals and pesticides. That extent, the environment is polluted. That extent, that means imagine the blood is having the residues of toxicants, in the sense, what is the metabolic aspects of organs, maybe kidney or brain or liver or other functionally different tissues, that fate of the tissues, imagine. That's why the person directly or indirectly, he doesn't know what happened for me. I am healthy, but how it has happened. This type of confusions are creating present day because of the environmental toxins which is accumulated in the environment. So here, the common disease, yes, I want to say a few examples for the typhoid. Typhoid is, you remember, this is entered through food and water only. If the water food is this type of typhoid, enteric fever, is a very common. Most of our drainages are not properly cleaned. Most of the potable water which is channels are in the drainages only. Wherever the breaks, that drainage water which is enter into the family's potable water, along with potable water people are consuming and causes this type of disorders. Remember, as the statistics says, one statistic says that is almost 80% of the people are not consuming safe drinking water in India. Not only India, the number 1 in the world is the Asian countries. If you refer the statistics, FAO statistics and WHO statistics, it's a shockable incident. These things, many people, though they know, but there's a bother. Anyhow, pneumonia, another type of disorder, which is Staphylococcus pneumonia, which is that is enter in the lungs related problems. This is also one type of drastic disorder. You can say how these pneumonia will attack especially in the lungs and alveoli. The water is fluid is filled in the alveoli, so that net releasing of oxygen required or purification of blood is very, very limited. As a result, supply of blood to the organs is very, very low, very, very low. So that's why this type of, you can see the system, the symptoms also I mentioned. You can refer this in this left side corner, how the lungs and primary bronchus, secondary bronchus, terminal bronchus, tertiary bronchus, alveoli, everywhere these epithelial cells and squamous epithelium are totally affected. As a result, chest pain, one type of chest breathing problems. Also, these are the problems what you find in one of the environmental disorders. Another one is tuberculosis. It is also in the environment very common. Very common. This bacteria, this is Macrobacterium tuberculosis. This also is relevant to the pertaining to the environment only. And in this slide, I can see, I show this variety of bacteria how is influenced, because this is my focus is hazardous chemicals, hazardous diseases caused through environment. If I want to explain this, every aspect, it requires so many hours to explain. Just for, lead you, bring you how the environmental toxins causes the hazards to the human being. This aspect is the major target of my talk today. So this is a pneumonia, so many bacterial meningitis, which is related to the brain. And this in the last sexual transmitted diseases and skin related diseases, urinary tract infections, gastric related GI, gastrointestinal tract related disorders, and upper respiratory disorders, and that is a nose and a lung related. These are the variety of bacteria which play, including the common cold also. There is a rhinovirus, the same such a category, and through the environment only spreads. And here we are not, we are much familiar with COVID-19. This is how the COVID virus, virus will acts in the lungs. I shown here, that is enter in the alveoli, where it creating a lot of abscess in the alveolar tissue, where the, purification of blood is affected. Along with that, other organs, like you can see here also, the COVID-19 damage affects the body, not only the lungs, other structure also, lungs and even the kidney also related. You see, this virus, which is a dormant, sometimes is a dormant stage. Virus is changing their morphological structures. Type to even today, the another form of virus is there, but they can change, they can modify, they can the genetic material of makeup of that virus is such a mode. They can change, they can duplicate, they can modify themselves within the conditions only. So here also the brain part, lungs part, this is also one of the other recently we come across in this type of problems. Other viral, in this slide I given so many viruses which is pertaining in the environment only, which is related in the environment. Through environment contamination only, this type of virus is acting on the body and cause hazard. Yes, here, one example I can say, here the leather and tannin industry, where they are using the animal skin and skin products by chemicals and other things. If you visit the places like in Tirupur, in Ambur, in Tamil Nadu, even there is the multinational companies are established the industries in that area. They people, the US and other people, they are investing the money for the factories, and they're taking the processed skin. And for the processing purpose, they're using variety of chemicals and other muscular and bacterial contaminants, etc., are loaded in the area. So that area is a highly vulnerable area. Recently, Tamil Nadu government is declared it is a vulnerable area for the such type of infectious disease like that. Why? Because this industry, the owners of this industry, they are highly rich people. They have the muscle power, money power, and political power, and they are not at all bothered about the regulations and rules, and including the ethics, and they are contaminating in the industry. Why not? There is a West Coast paper mills in the Kauli River. There are 340 micro and mega industries are established in that basins of the river. Their releases, including the molasses, I'm saying, and they are entering, and that entire Kauli River is today is become a worst condition. I studied, even the factory people are not allowed to take the samples. They threat us. That type of muscle power, and if anything is come out, the people are get unpopular, etc. These things are going on. So this is why even the paper mills, effluents, they containing maximum this type of viruses and bacteria, which one way or other, the people are in and around that area, definitely they contaminated, they cause the touch disease. That disease infect one person to another person and transmitting to area, city to city, state to state. And here malaria also one such problem. We know very well about the malaria, which is a carrier of a mosquito, female Anopheles mosquito. This mosquito, if you see the life cycle of the mosquito, this mosquito will grow where the water is stagnant, where the water is ugly water, stagnant water. They improve their life cycle, and they complete the life cycle. So this is also, even we used to educate, we should not allow the water in under our premises, etc. But people are not bothered. Why? I don't know. whom can take action, we don't know. But even inside, children, like this, the people are thinking and neglecting, so that this type of dreadful disease we are facing. Other aspects, apart from the bacteria, virus, and protozoans, another problem is parasites. Parasites is a very, very bad disorder today, what we are facing. I mean, amoebiasis, including from the amoebiasis to particularly endoparasites, pinworm, seatworm, hookworm, roundworm, pinworm, seatworm, hookworm, roundworm. You might have seen that is called Taenia solium, Taenia saginata, beef tapeworm, dog tapeworm, because the sanitation in our country is totally unhygienic. Even in spite of that, the government is announcing cross together to construct the toilets, fresh out toilets in the villages. The people are misusing. I cannot, please don't think I'm blaming the government. The setup is like this. And the people are excreting the excreta in the roadside, other side, where the flooding time or rainy season, these excreta contain the spores of these parasites enter in the drinking water point or ponds and lakes or etc. nearby. The people are use that water for different purpose. Automatically, this type of parasites enter and causes havoc. It's a silent killer, what I'm saying. Okay, like this, you can see river blindness, one type of high worm. So here the parasites mold. If you study the life cycle of the parasites, especially the roundworms, endoparasites, intestinal parasites, they migrate towards sometimes the intestine, from the intestine, small intestine to large intestine, along with fecal matter, they come sometimes. Sometimes they go reverse. They enter in the bloodstream, they reaches to the brain. They causes a brain-related damage. They reaches to the eye. They cause vision-related problems. Sometimes they enter in the lungs and causes respiratory-related problems. And sometimes even you might have experience some people in the villages and all. The worms come out of the mouth also. Because this type of phenomenon we are noticing because of the contamination, etc. Ascaris lumbricoides, which is called as roundworm, Ankylostoma deodorale, hookworm, and tapeworm, Taenia saginata, Taenia solium. These are the very, very popular, very, very common, very, very contaminated, very, very dangerous disorders. Yes, another thing is I come to these are the biological means. What about the chemicals and other substances which is persisting in the environment? The persisting in the environment, chemicals. Today the industries or there are n number of industries, textile industry, petrochemical industry, or leather and tannin, sugar industries, so many industries, they're releasing one way in the leaching in the way of effluent, another way is through the chimneys in the form of gas. So these synthetic chemicals, these chemicals which is persisting in the environment, maybe aquatic or maybe terrestrial or even aerial, sometimes they react themselves and form a secondary and tertiary products. The secondary and tertiary products are so dangerous than the parent compound or parent element, like that the research says. So best example I will tell you, endosulfan. Endosulfan is a very common insecticide used in the agriculture. All food crops are edible crops. Food crop, including vegetables, etc. Even the commercial crops like cotton and sugar, etc. They used. So once the endosulfan enters the organism's body, they convert it, they mix with the other molecules present in the intestine or etc. They develop into endosulfan sulfate. This endosulfan sulfate is 25 times more toxic than the mother endosulfan. So outside the environment, it is a ordinary endosulfan only. Once it enters in the sense, the virulence, the toxicity is become multi. As a result, the organism is affected very drastically. I will tell you the magnification process in coming slides. So these are the chemicals are classified, the chemicals which is persisting in the environment. If you want to read, there is a book called as Environmental Science by Cunningham's, that book you refer, they give a lot of information about the environmental toxicants. So here, these chemical agents, there are two types, that is hazardous chemicals and toxic chemicals. Hazardous chemicals and toxic chemicals. Here they're listed out like this: flammable, explosive, irritants, sensitizers, acids, and caustics. This is a one category. Other type of chemicals, they are called as a silent chemicals, and the virulence is very high whenever the chance comes. For example, I can say the DDT. DDT is the half-life period is 34 years. 34 years is persisting in the environment. If you suppose the DDT is used in the soil where you are cultivating some edible crops, definitely the DDT is entered in the foliage, plant parts, maybe leaf, fruit or stem or flower, etc. We are consuming that. Definitely that DDT is entered in our body. So the half-life period, since it's 35 years more, it is persisting in our body till the denature. That means if you are adding this every week or every month or every year, the magnitude, I will tell you about the biomagnification process in coming slide, and automatically the outbreak will take place. When organism's threshold level crosses its limits, the organism will fall. The failure of the organs, maybe kidney or maybe liver or maybe heart, etc. That's why here, these are the listed chemicals which is highly persistent in the environment. Many people are not bothered about whether it is there or not. Some people still suspect, yes, these are the chemicals present in high concentration. And this, even though once or two times, 1 week or 1 month, 1 year, if you expose this chemical, doesn't happen. After 30 years, after 40 years, after 50 years, after 60 years even. The susceptibility depends upon the immunity of the individual. Susceptibilities depends upon the immunity of the individual. So that's why the expression it takes some aspects immediate, they take long. Most of the xenobiotics which is present in the environment, they have the long off-life period. Anyhow, that's with the flammable chemicals. Just a glance for important one, that is camphor, cellulose nitrate, and naphthalene. These are very commonly used in our houses. Our domestic level, they are using. So here, this property, these are the decaboron, lithium azide, phosphorus, heptasulfate. These are invisible substances. Even it is there, we are not much concentrate, much bother about it. Nothing will happen like this. But definitely it will happen. It has its own hazard. That is a simple one. You take the camphor. Camphor is used, I list out so many ointments where the important ingredients. If you expose these camphor regularly, definitely after 3-4 years, that side effect will be seen. These are, you see, the composition, what we are using very regularly in this, but this type of problem. The camphor is ingested in the higher accounts, unsafe. I given some point, the skin-related problems, burning of mouth, throat, nausea, vomiting. Especially children are very much prone to this. While doing this type of chemical, please don't expose the children. Many times we are, by the ignorance of this, we are into, because children's skin and body organs are very tender. They recognize, and easily they respond. But we are thinking some other reason maybe, But that type of things will accumulate and finally cause a very big hazard. This is a cellulose nitrate. This is an amalgamation of acids, nitric acid and sulfuric acid, etc. They are used in many aspects, many domestic uses. They are very another very toxic substance available in the nature. Next, they are easily fire, intense flame, and poisonous. They will act different manner. And here naphthalene balls. Naphthalene balls is also, we are in the houses, we are using this insect free. Our cockroaches should not come like this. Though, of course, by the smell of the cockroaches may not reach, but it's a highly toxic. The fragrance purpose. Not only this naphthalene, many room fresheners, very dangerous, very dangerous. Sometimes you might have experienced continuous irritation in the throat, or continuous sneezing, running nose. That will cause us, they are the alarming symptoms. If you expose continuously, definitely that will cause a disorder. Yes, it's used in household, mainly used for the fragrance purpose, keep away cockroaches. Ingestion leads to, hemolysis. That means sometimes contamination. Sometimes children used to have etc. The incidents are there. The main reason, as a research says, the hemolysis. What is hemolysis? RBCs. We have the red blood corpuscles, which are carrying the oxygen as well as the nutrients to the cellular level. So here the viability of the RBC is reducing. The size of the RBC is reducing. The carrying capacity of RBC is reducing. Oxygen holding capacity. When it binds with oxygen, it became oxyhemoglobin. This oxyhemoglobin is a highly essential component for the cellular respiration. Wherein this RBC is lost its viability, lost its function. Once its carrying capacity is less, the ATP production, I mean metabolism process is diminished or reduced. As a result of reduction of the metabolic profiles, the ATP, cellular currency, what do you say? These will be, quantity is less. Once the ATP is less, the organism's functioning phenomenon is automatically less. He is always want to bed, unknown fatigue, a body ache, muscular pain, he wants to giddiness feelings. These type of things will create. This is, we are experiencing many times, but why it is experiencing, what is the causative factor, we are not at all bothered. If it crosses our limit, we will approach the doctor, they will give some tablets or etc. It's temporary relief only. So remember, this is another very drastic component present in the environment. Explosives. We don't know any words to talk about the explosives. In the beginning, when I told, the many countries are competent about the war weapons, production, managing, etc., etc. These explosives, ammonium chloride and barium azide, RDX, INT, this nitrotoluenes, aromatic compounds. Aromatic compounds, particularly we are using the fragrance purpose and other scent and other things, what you call body odors, etc. Please don't use it. Don't use it. It's a dangerous. In the beginning, after using, simple thing, I can say one word. There are so many, today the era of makeup, cosmetology, cosmetics, talcum powders, vanishing creams, fairness creams, fairy ever, lovely, this and that. This, if you see the functioning phenomenon of the fair ever or fair and lovely, etc., etc. If you see the biology, in the skin we have the melanocytes. In the skin there is a melanocytes are there. These melanocytes which is grouped, the person looks like a dark complexion. If the melanocytes are scattered, the person looks very fair. This is a common way. These chemical, what we can say the vanishing creams or sunshine creams, aging creams, now it's very popular, very costly also. It's a very, we don't know what type of chemical we using, them listed the names. You refer the each individual composition of the chemical, it is a very dangerous aspect, you could come to know that. Yes, these chemicals, what to do, say in a fair ever or fairness creams, after applying this, temporarily 2 to 3 hours, that grouped melanocytes are scattered in the skin. As a result, you will feel a freshness. Often you will look at a mirror and I am so fair like that. You feel like this. After 3 hours, automatically the disturbed melanocytes again come to the original position, again black only. So for this, there are so many other facts which is involving. There is even the hormonal function also involving there directly, indirectly, like that says. That's why this cosmetology, powders and etc. Are very, very dangerous. For that, go for the eco-friendly. Even nowadays, people are talking about many things about the eco-friendly. I don't know. Even funny thing is, these people, if you are not become a white or fair, your money will be returned. Advertisement, right? Yeah, how many of us we are returned the fair and lovely and get the money back? How many of you? Come on, tell me. It's not possible. This is a bluffing us. We are bluffed. And like this, there are so many such bluffing actions are there, and I will tell you if time permits. Anyhow, these explosives also very dangerous. Even we are experienced well about Hiroshima, Nagasaki and all. Radioactive isolation, radioactive isotopes, lithium 90, strontium 131, radon. These chemicals are very commonly persisting in the environment. These chemicals are regularly using in the war related weapons manufacturing industries. Due to the smelting of these radioactive substances, along with the smoke and grit, they go away in the atmosphere and fall. Chernobyl incident is the best example for that. These strontium 90 which enter along with the food, that will interfere in the bone marrow. Bone marrow, blood RBC producing phenomenon is reduced. That's why people become very, very weak, anemia. How do you test this? Because after so many tests only, we come to know that this is this. Because ordinary tests, it's not possible to detect. Today, most of the toys, most of the other decorative items, they are used one way or other, this type of materials, and it's entering in this environment. Next, ammonium chlorate, yes, hemolysis, methanoglobemia. Methanoglobemia means became a blue color. The person became a color change. Color change, a complete blue color alla. Not that blue, complete blue color. Actually, the person is very fair, but due course of time, you will become a complexion is neither black nor fair. Black, is not become a complete black and some other different color. This is called as methanohemoglobemia, that type of coloration. Next, renal damage. This is another very, very pathetic disorders what we say observing present day, including teenage people to up to the old age people. These renal related problems, kidney related problems, kidney swelling, Bowman's capsule, shrinkage of Bowman capsule, glomerular dysfunction of glomerular function, reabsorption of nutrients, and like this variety of way. The urology department, they are busy now. Next to the cardiology, now the urology department is very busy because of the people are suffering a lot, this type of disorders. And barium azide. He says causes respiratory problems, barium azide. Yes, burning sensation, induced, heavy breathing, and other symptoms like nausea, vomiting, diarrhea. These all are headache, dizziness, temperature, temporary vision loss. These things are very much seen in the people. Nowadays, it is no need of explaining because of its use for the weapon, for bomb and other things. This is yellow-colored trinitrotoluene. Chemistry people understand very aromatic compounds, synthetic aromatic compounds, particularly, though they give a good fragrance smell and all, they causes variety of disorders. They use the major raw metal for the manufacture of explosives. Irritants, yes, this is also, whenever we visit some other Delhi and other areas, highly polluted areas, automatically we will feel some type of nausea, vomiting sensation, one type of nose irritation, one type of eye rubbing nature, eye irritation, etc. This is because of there are so many irritants are persisting in the environment. I will tell you, this irritant, particularly one type of irritant, silicosis. Morning lecture also one person is told about the silicosis from the the grasses of paddy, etc., etc. Yes, here the people who are working in these silico related companies, they develop one type of disease called as silicosis disease. Yeah, silicosis disease is very dangerous. This silico substance enter the lung, they make lung become a puncture. Lungs become whole. That's why they call it as silicosis. The lungs become whole in the sense, imagine, the lungs get punctured in the sense, what is the fate of the individual respiration, etc., etc. Imagine, one type of nasty, persistent cough, dry cough, continuously the person is suffer. This is the persons who are silicosis, black lung disease. Black lung and white lung and brown lung. These are the people who are suffering where the areas, highly industrial areas. Cement industries, glass industries, asbestos industries, or even the cotton fiber industries, cotton yarning, cotton mill, textile industries, where enormous quantity of dusty soot metals are entered in the atmosphere, and the people are respiring. These tiny particles come and settle in the alveoli. As a result, what happened? The alveoli is a very much, elastic property. Whenever the gas comes, they can bulge, and when gas come out, inspiration, expiration, they suppress. This elastic nature of the tissue is lost. So this contraction, relaxation property, once it lost in the sense, the intake of atmospheric gas, oxygen intake to the alveoli reduce. As a result, the lung tissue become rough and hard. As a result, automatically, net respiration is automatically collapse. This is a very common scene in the industries where cotton industries, etc. You can see the silicosis, the figure, you can see the holes, round holes, no? The white lung, brown lung, black lung, the lung became black. Especially people who are working or people who are living in and around the coal areas, coal factory, coal industries. Yes, the method, how it is works, how to, I will leave this slide here. Anybody wants to can take it and you can read afterwards also. The mechanism of how the lung, respiratory failure, dysfunction of lungs will take place, etc. given in the systematic manner in the illustration with figure, especially, conjunction type mining industries. What is in the left side, there are the different disorders also. Particularly, yesterday only I referred in the newspaper, the people working in the paint industry, paints, oil paints, and Xerox machines, ammonia print, etc. They are suffering a lot for this type of disorders. Yes. Benzogonium chloride. This is another one type of toxicant which is persisting in the environment, which causes the immune-related problems, immune-related neutrophils, basophils, eosinophils, white blood corpuscles, you might have seen, RBCs and WBCs, hematology aspect. They suffer a lot from this benzogonium chloride. And here, this is lung, I told, not just now I told, coal industry, if you observe the lungs in such manner, and brown lung disease, this is also one type of disorder related to cotton industries, ironing industries, textile industries, where this type of disorder is very common. But even though people are, industries are given a proper instructions, you have to filter the gas and release. But no industry is following, because that process is very costly. That's why the industrialists, they are not opted such type of technique. Purification the gas and release. That time, simply whenever the dark time or whenever the night time, they release the gases. Even the water also, such things happen. Here, asbestosis, asbestos sheets in the cement industries, glass industries, such places, you could find this. And sensitizers, yes, allergens, and agenopause metal, that is one type of highly material which is carcinogenic. Some chemicals silent, in the beginning I told, no, carcinogenic, radioactive, and developmental toxicity. Developmental toxicity in the sense, embryo doesn't grow. In the recent, NIN is one type of National Institute of Nutrition, one incident. Recently, the one type of limbless babies. The babies born with defects. That's called as a teratological disorders, embryological disorders. This is very common. Amyl acids. This is acid. This is the only country like India, acids are available freely, because the miscreants, the criminals using this acid in different purpose. This is the incidents of the people who are thrown this acid in the faces of some people, etc. But there is a regulations. Acid should not sell wherever, whenever, whatever. Without proper license, etc. But this is also misusing acids and caustics and mutagens. Some are mutagens, DNA, teratogens. Just now I told the teratogens. Here, teratological aspects. Now in India, it is a very limited, but in the European countries, this is a very dominant, especially people consuming alcohol. Especially women folk consuming alcoholic beverages, that alcoholic content is persisting in the body when the embryo is growing. During that period, and the embryo will get affected. The limb formation, organization of organ system, the embryological aspects, you could find this type of disorder. And carcinogens and asplicians. This is also one type of the chemical which is released. There's two types: active asplicians and passive. This main characteristic feature of this chemical is reduce the oxygen level in the atmosphere. So oxygen is a particular percentage. If these chemicals are entered in the atmosphere, the oxygen level is reduced. How we are facing today, the carbon dioxide level is increasing, but these chemicals, they are reducing the oxygen level. So net availability of oxygen level, 21%, that percent will be decreased. As a result of so many ventilation problems, because oxygen is a living gas, and because of that gas oxygen deficit, the organism will die. And toxins, here again I will come to the toxins. That's the chemicals. Other things, toxins. Toxins are two types: natural toxins and man-made toxins. If the toxins which come from the animals, study is called as toxinology. Toxicology is nothing but the study of chemicals which is come from the industries, etc., etc. Remember, toxicology and toxinology. Here, the toxins are two types: synthetic toxins and natural toxins. Natural toxins available in most of the animals, even cones, one type of molluscan, or one type of spider, one type of some type of snakes and all, adder snakes, etc. They produce a very toxic necrotizing agents. Once the toxin is fall on the body, the tissue is collapsed. The tissue is putrefied. That type of necrotoxic substance also produced in the natural toxins. And yes, I will show a few toxins. Natural, synthetic, and natural toxins is biotoxins is called as a biotoxins, particularly cones, Anhydrina hydrophis, sea snake, Anhydrina hydrophis, very dangerous venom, which is a very portal. And even these are the spiders, snakes, scorpions, jellyfishes, etc. They are producing, and necrotoxin, just now I told, one type of snake called as adder snake, which is produce one type of peculiar chemical present in the poison. The poison is nothing but a protein only, and these particular adders, this enter in the bloodstream, they enters in the tissue level, the tissue get putrefied, and this is the red-billed black snake. It's a very dangerous snake which is producing that particular venom. This is the adders, one type of Russell's viper species. They cause puff adder, very dangerous. Of course, it is it's not at all seen in India, but other countries it is very dominantly seen. Some of the spiders also such categories. And here I want to say giving some flesh eating, some bacteria. Here in the environment is in the Africa and some other Asian countries, very dominantly seen particular bacteria. One that is necrotizing facilities, one type of name of the bacteria, which is spread through the air only. This bacteria is located in the fecal matter, dirty animal resources. Yes, in such places, this bacteria will come and that enter. That's why this is another. Yes, you can see. Just time factor. I can. Yeah. These are the other than the things. 5 minutes, last 5 minutes. The pesticides and all. You know the pesticides. There is no word to talk about. Organochlorides, organophosphate, and carbamates. Without these three chemicals, no pesticides at all. And highest production of pesticide industry is there in Asia only. Highest consumption of pesticide high in the world. Pesticides only. And these are the different pesticides, organochlorides, organophosphates, and the mode of applications. It's become a big nuisance. And even this is the one thing to understand. One, the toxic and nothing but pesticide, one delivery, they interact with the target molecule and enter in the cellular level and spoils the cell and dysfunction and despair means collapse. And these are the variety of different types of pesticide, DDT, I told. And here one thing, the endosulfan in the Kerala, where the people are using 20 years, all of a sudden outbreak takes place. Even you can see the people there in the one village called as a Puliyur and Padri area. These are the people still they are suffering. Hydrocephaly is a major disorder. And yes. This is the environmental components, these are the major way of transforming the xenobiotics one trophic level to another trophic level. Biotransformation, bioconcentration, bioaccumulation, biomagnification. Any xenobiotic, this is the only process enter the biological system, and you can see the reproductive failure, immune system. So what something is open? Yes, in this slide, out of food chain, anywhere we will be there. Osprey means is a tertiary carnivore, where human beings also we can place there. And there are so many information is there, and you can go through. This is another experiment we did in my laboratory, and we are using the chlorpyrifos with a 0.2 micrograms per liter by micro drilling the egg and introduced and incubated, and after 21 days we find this type of anomalies in the body and the growth factors of the chick. So the first one is a control, remaining all are affected, different intervals, different dose and different period. This is also embryonic. The first is a control one. These all are affected one. Like this, we can see so many experiments we find, and in the research we can find there are the slippery tendons, and in the chicks we work up to 6 months, from the day one chick to up to 6 months with the constant supply of the pesticides and all. These are the effect. These are the different titles I given. The slippery tendon, chondrolithocytosis, like this. They are this is one type of paralytic stroke the chicks develop, and and you can see the partial paralysis, one leg with totally paralytic, and how the limbs and other things will happen. This is the X-ray studies. The center one is the healthy one, and both the sides is contaminated, infected with toxins, various toxins, and X-ray and bone formation. And even the first one is affected one, and normal is straight. Even the bird which is feeded with the toxins, they grow very elaborately, very fast. But there is a deformities also we could notice. And this is the heavy metals. I don't know, this is a Manamuda part, and this is a heavy metal toxicity, and causes variety of disorders, heavy metals, chromium, yeah, how it enters, the symptoms of mercury poisonings, and this is the way, how it is a different way the heavy metals impact, heart, lung, liver, and different, even the reproduction part. Even etc. Even this is the symptom of chromium. This is very common, especially the chromium toxicity, very much outbreak in Orissa recently, where the mining industry is there. Even the Ballari area in Karnataka also, there's alarming variety of drawbacks, including the genomic level, chromosomal level, the toxicity is work. The DNA complements are also changing. Purines and pyrimidines, DNA base pairs and pentosugars, that level is altering. And you can see the adults and children's about these heavy metals, and additives, finally food additives, people are attracting towards the restaurant food and all. They are using very, very bad. I'm using a sodium nitrate and profile gallate. These are the highly toxic substances used for the preservative purpose. These are the different effect. After 6 hours, after 1 hour after, these are studies, WHO study report, and these toxins, colorative substance and artificial sugar, sweeteners, these all are very, very dangerous, and even particularly potassium bromate. It is very much prevailing in India. Bakery food, they are much used in the potassium bromate. It is a very, very toxic. And even the using, baking soda, baking soda, idli doses and all, the softness they get. But it will affect the villi. It will affect the villi. Many people are suffering indigestion problems, because you feel some smoothness and whitish color, that is because of that baking soda, etc. Another thing is sodium benzoate. Sodium benzoate in the pickles and all we are using now. The pickles is half-life period is only 3 months or 2 months. But 1 year, 2 years they are maintaining in the sense because of sodium benzoate. Sodium benzoate is a very, very neurotoxic as well as hemotoxic. It alters most of the digestive enzymes. That's why indigestion is a major drawback, what you see. And these are the food colors, and you can see how that one each wear. I put one color. These are the popular colors using in the restaurant foods. That's why be alert. And because here the problem is, there is another hazard. There is a hazard. How to protect, how to maintain, how to monitor, how to conserve is a big question mark. For that it requires a lot of efforts, lot of research, and lot of education. Awareness is needed. Thank you. Okay.
Thank you, sir. Now, I would like to request.
Any questions in this regard? Can you clarify? No problem.
Any questions? I think that there is no question. Okay. So I would like to invite Dr. S.M. Gaikwad, sir, for giving a remark on the speech. As well as I will like to request Dr. Sujata Patil ma'am for vote of the thanks.
Actually, we are running late, so I will not take your more time. So to summarize his lecture, it is a very difficult task for me. Notably, he explained the effect of toxicants on our health. As towards the eastern part of this Islampur, there is a grape fields, farmers. So miscarriages are the very serious problems in Hyderabad and Secunderabad area due to the application of pests on the grape fields. So we have to think on this. So pesticide application must be reduced. Actually, pesticides are must, but we have to reduce its use, and we have to find out the remedies or the replacements for this. So, sir, with your very thought-provoking lecture, you have touched all the diseases which are occurring due to the toxicants, and I hope from your lecture, especially the young generation will take home message to reduce the toxicants, so that, for example, we can reduce the use of plastics, we can reduce the use of pesticides, we can make awareness among the farmers to reduce the use of pesticides. So even the day-to-day life, we are using the camphor, naphthol balls, and the room fresheners. So this is also very dangerous for our health. So many applications and their effects on our health, sir has explained. So thank you very much, sir, for your thought-provoking lecture. So I understand that you will need more time to give the details about these toxicants and their impacts on our health issues. Thank you very much. And finally, I thank the organizers, the convener, and the society authorities for giving me opportunity for the chairing this session. Thank you very
sir.
much.
Thank you, thank you.
Hello, good afternoon. I know it's too late for the next session, so it's my duty as a part of this occasion to express vote of thanks. Now this session, it gives me immense pleasure to express vote of thanks Dr. David sir for his very inspiring and eye-opening, or I say thought-provoking lecture. He delivered very nice lecture, so thank you, sir. As well as I am also thankful to Dr. S.M. Gaikwad, sir, for chairing this session and giving his remarks. I am also thankful to Dr. Gezge, sir, for working as a coordinator for this session and giving brief introduction of this guest. So, special thanks to all the audience, researchers, and all my colleagues and all everyone present in this hall. So thank you once again. Thank you very much.
Thank you, ma'am. So again, immediately we are going to start oral presentation session.
1 minute. In this moment, I thank the authorities, particularly friend Gejge, and I have associated with him from long days. 2007 also I came in this conference in the same hall only. I once again thank all participants and delegates and authorities. Thank you.
We are too much appreciated by your lecture. From Department of Microbiology, I am Anil Salunke and Swati Kulkarni.
Thank you.
We are too much appreciable to your lecture because of your giving very nice information about diseases.
Thank you. Thank you.
So immediately after this, we are going to start oral presentation. Life sciences oral presentation is going on in this hall, and physics, chemistry, computer science and mathematics oral presentation will be arranged in virtual conference hall, which was next to this hall. Those who are interested to give oral presentation, one information I want to give to you about time limit. Only 5 to 6 minutes which was provided for each oral presentation. A lot of oral presentations are there, that's why time limit is there. Only 5 to 6 minutes which was provided to you for oral presentation. Okay? Okay. So, for oral presentation, chairperson is Dr. D.B. Nakade, sir. chairman of BOS in microbiology, Shivaji University. So, sir, you take chair for that program. Then chairperson is Dr. M.R. Abdar, sir, KNP College, Valava. Examiners are Dr. Rajendra Laute, sir, from ZAT. And next examiner is Dr. M.B. Wagmare, New College, Kolhapur. Coordinator for this program is Dr. S.A. Kulkarni, ma'am. And rapporteur is Y.D. Kengar, sir, Dr. Y.D. Kengar, sir. So, the list of oral presenters are here. There are about 32 oral presentations which was going on. So, the first oral presentation which was given by G.B. Kadam and Dr. S.R. Rangdale. I request Dr. Swati Kulkarni ma'am to introduce Dr. Dhananjay Nakade, BOS chairman in microbiology, Shivaji University.
Good afternoon, everybody. Now I welcome all the participants for oral presentation which are assembled here after lunch break, and the very good presentation made by Dr. David, sir. I am going to introduce our chairperson, Dr. D.B. Nakade, sir. He is professor in microbiology at Rajaram College, Kolhapur. He has teaching experience of 29 years and 3 years administrative experience. He is recognized PhD guide of Shivaji University, Kolhapur. Under his able guidance, these students have achieved or pursued PhD degree. He is coordinator of Virtual Learning Center at Rajaram College, Kolhapur. He has been awarded with Fellow Award of International Science Congress Association in 2011 and Excellence Award for Organization of State Level Competition in Life Sciences organized by Science Association of Rajaram College, Kolhapur in 2013. He was awarded with the Best Microbiologist Award, Microbiology Society of India in 2014. He has also won Best Paper Presentation Award in International Science Congress Association 2014. He is also a principal investigator of the project entitled with production and characterization of antifungal compound from Halophilic Bacillus subtilis Na17. He has participated national and international conference seminar symposium and presented research papers also at the conference. He is now selected as a chairman of Board of Studies at Microbiology at Shivaji University, Kolhapur. So I am very proud and I am very happy to have such type of guests and eminent personalities among us for the paper presentation as examiner or chairperson. And I also welcome all the dignitaries, including co-chairperson as well as the examiners, Laute sir, as well as the Abdal sir and Wagmare sir on this dais. And I request the examiners to judge properly all the papers presented, and I also wish that you will enjoy definitely such a academic journey at this hall, and I give all the students best of luck for that presentation. Thank you very much.
Thank you, ma'am. I would like to request convener of this conference, Dr. S.R. Mani sir, to felicitate our invited guest. Dr. D.R. Nakade sir, D.B. Nakade sir, which was felicitated by our convener, Dhananjay Nakade sir from Rajaram College, Kolhapur. Again, I would like to request S.R. Mane sir to felicitate Abdar sir. Again, I request Dr. S.R. Mane for the felicitation of Laute sir, Rajendra Laute sir. So, I would like to request our principal, Dr. Vilas Kale sir, to felicitate our examiners for oral presentation. Dr. Rajendra Laute, Also, I request Dr. Kaya sir, please, to felicitate Dr. M.B. Wagmare sir from New College, Kolhapur. So thank you, sir. So here we start oral presentation. There is a time limit, please. So the first oral presentation, which was the participant is Kadam, GB Kadam. So all oral presenters must be report here. So I would like to give you one information. At first you introduce yourself, your college and name of the topic. Okay? And the time limit is 5 to 6 minutes. After 5 minutes, completion of the 5 minutes, bell is ringing.
Okay, sir.
Okay.
Good afternoon. Good afternoon, one and all. First of all, thanks to the organizer committee given me this opportunity. Myself, Geeta Bhagwan Kadam, Anna Sahib Aute College, Manchar. Here I am present for the review article, that is a review article on the Tyloflora indica. Abstract. As you know that India is the rich diversity and there has the too much medicinal plants is there, as like Ayurvedic, Unani, Sindha and the medicinal plants also. So India in use as so many medicinal plants also. So here Tyloflora indica also so much important plants we have to use here, and it's also known as the Tyloflora indica is known as a Antamul. Anantamu is the Hemidesmus indicus is that the different, but this the Tyloflora indica it's known as a Anantamul. It's also known as a Khandari, Pitkavadi, Pittamari and Dhammakavadi. Taxonomically, Jagtap and Singh, this the researcher, they are the reported that 21 species and the 2 varieties of this Tyloflora. This genus in the Maharashtra, 6 taxa is there: Tyloflora indica, Tyloflora dalzeli, Tyloflora rotendifolia, fasciculata, and the pubescens. This plant is used as a medicinal. This not only the leaves, roots, and stems also. Roots of the Tyloflora indica possess the stimulant, emetic, catherating, explorative, diflorating, and it has a good natural preservative also. Leaves and the roots, they are used as a substitute for the root drugs. It is used in the Brazils. And it's also reported that the uniforms and further medicinals also. These plants are the too much importance in medicinally. And this scientist is there. They reported that this Tyloflora indica used as a asthmatica. Introduction. Tyloflora is belong to the family Apocynaceae, first Asclepidaceae, now this thing to the Apocynaceae. And this Tyloflora, again this name changes also, Vencetoxicum. So this Tyloflora is the clamber, slender, and another species is there, Tyloflora dalzeli. They have the same medicinal properties, but till now not their constitutes the known. It's also known as a folk remedy, dysentery, diarrhea, asthmatica, and most important constitute of this Tyloflora indica is the tyloflorin. Significant activity of this stem and leaf extract they have to use for the cancer's activity also. WHO also reported that the 235 million people they suffering from the asthma, and they also tell that this crude drug is the beneficial for the asthma diseases. It's also necessary for that this alternative source for the Tyloflora indica for important and the very much useful for the asthma diseases. There are the 3 species is there: Tyloflora indica, rotendifolia, fasciculata. It's much the indigenous to the indica, and there are 2 varieties also. They are the found in the India also. Here that some taxonomic groups. Tyloflora indica is belong to the family Apocynaceae, subfamily Asclepidaceae. Here useful is the asthma. It is a Tyloflora indica flowers, and second one is the Tyloflora fruit. It's belong to the family Apocynaceae. It's the possess as a biological activity also. Taxonomic, botanical distribution and the descriptions. These are the again climbers, and they are the, it's known as it twins also. They are the too much small, like fasciculata is the climbers, and again this Tyloflora indica is climber is nature. It's found in the Himalaya tract, peninsular India, eastern, northeastern. In Maharashtra also this plant found in the Pune, Marshes Ghat, Lonavala, Khandala, this species also. This Tyloflora indica is the branching climber, shrub, and their leaves are the ovate to the oblong in natures. And their calyx corolla as like a fayu, they belong to the family Asclepidaceae. So just like a Calotropis, everyone know that the Calotropis. So this plant also belong to the that family. Fruits are the follicles and seeds are the Eteroberry. So their size is the 0.6 to 0.4 in long. This plant shows the some phytochemistry, and these are the too much importance: alkaloids, septenins, ABCD, EH, IGs, and these are the non-medicinal. These are possess the primary metabolites and the secondary metabolites. They presents in that. So because of this phytochemistry, this plant useful as a folk remedy and the traditional use. Most of this plant use as a asthma in nature, asthma. So because of this plant shows as anti-cancer activity, antioxidant activity, anti-asthmatic activity, anti-allergic activity, hepatoprotectic activity, and so many researchers reported is that. So here now I am here just I am summarize all this activity in this review article. Now I am conclude that this Tyloflora indica, not only the Tyloflora indica and also two varieties also. It's the too much important plants, and they have to the useful in nature. Here I am collected some references, and thank you all of you. This all 4 species of the Tyloflora indica. Okay, thank you.
Thank you. Hello. Any questions? Okay. Next participant. Okay. Thank you very much. Okay. OPA1. Oral presentation which was now carried out, which was OPA1. Okay. Now next oral presentation, OPA3. Shendge VM and Mane KK. Hello. The next oral presentation which must be carried out by Shendge VM and Mane RK. Mane KK. So which was not present here. Now OPA4, PR Mane, J Swami, CB Sarunkhe and GG Poddar. OPA4. A4 na? A4 na? Okay. Your time starts now.
Good evening everyone. Myself Pooja Rajendra Mane from Department of Botany, Krishna Mahavidyalaya, Budruk. I am pursuing my PhD under the guidance of Dr. G.G. Poddar sir and Dr. C.B. Salunke sir. Today's topic of my discussion is the genus Botryocloa from Peninsular India. Earlier, this genus Botryocloa is embedded in another grass genus, that is genus Andropogon, which was described by Linnaeus in Species Plantarum. Then later Vontinus recognizes section Amphilopis from this genus Andropogon, and Haeckel elevated it as a subgenus Amphilopis. Then Otto Kuhn in 1891 described the genus Bothriocla on type species Bothriocla anamitica. As I told, it was described by Otto Kuhns. In taxonomy, each and every species name is having some etymology. Then for my species, genus Bothriocla, etymology is, in Greek, Bothrion means small pit and Chloe means a culm. This genus is diagnosed by the special characters. This is having a special type of inflorescence. It having a central axis on which is surrounded by the several many racemes. Each raceme has more than 8 joints, and canaliculate character on joints and pedicel is the major character of this genus. Here we can see the diagnostic characters of this genus. This genus is having pitted lower gloom of spikelets. Then many racemes are arranged on a central axis. And this canaliculate character means this, in last photo you can see the translucent middle line on joint and pedicel of the spikelet is present. In field, this genus is quite confusing with another two genus, that is Capillipedium and Dichanthium. Bothriocla and Capillipedium having similarity in canaliculate character, but they are differ in having racemes more than 9 joints in Bothriocla and racemes having joints less than 9 in Capillipedium. Also in Capillipedium, spikelets present on tip of the capillary-like peduncle of raceme, while it is absent in Bothriocla. Also, Dichanthium and Bothriocla are similar with the inflorescence morphology, but they differ in that canaliculate character. Here these 3 genera showing different characters. This first one and last one are the showing that Capillipedium is having spikelets on just tip of the peduncle of that raceme, and in Bothriocla it started just above from the peduncle. In Dichanthium and Bothriocla, we need a special lens, that is 10x or 20x lens, to see that canaliculate character. Material and methods, I have collected all species from India in each and every season. I have prepared herbarium sheets. I have identified that specimens by using various grass literature. Then in India, we are having in world we are having 37 species and 4 subspecies, and in peninsular India we are having 13 species of Bothriocla genus. This is Bothriocla bladi, which is having lower raceme shorter than the main rachis. This is sessile and pedicel spikelet. The pits present on the lower gloom of a sessile spikelet. This one is a compressa. Compressa is an endemic species. It is endemic to Maharashtra. Some people are reported from Tamil Nadu, but that is a doubtful record, as I could not trace any specimen in field or in any herbaria. Bothriocla eversiana is recently described from India by Landge et al. Then Bothriocla glabra, earlier it was in bladi, but now it is distinct from bladi. Bothriocla grami is also endemic species from Amarkantak hills of Madhya Pradesh. Then this is very, very beautiful species of this genus. This is Bothriocla insculpta, which is having 2 or more than 2 pits on lower gloom of a pedicel spikelet. This is Bothriocla ischimum. Recently we recorded it. It is as a new addition to the flora of Eastern Ghat. Bothriocla jaini also endemic to Maharashtra. It is present in very high elevation like Kalsubai, Mahabeshwar and Harishchandragarh. Bothriocla kunziana is having lower raceme longer than the central axis. This is Bothriocla longifloea endemic to Tamil Nadu. This is pertusa, we can found everywhere in India. This is quite common. This is variation in Bothriocla pertusa. This is Bothriocla cediusimum, that is pruneous kind of nature, which was present on abaxial surface of the leaf. Bothriocla woodrowi, it is a very special species of this genus, as it having camphor-like smell, and it is endemic to Maharashtra. We reported from it Telangana and Gujarat, but population of Maharashtrian Bothriocla woodrowi is having strong camphor-like smell. As I described, we are having in India 13 species, and out of which 5 are endemic. Compressa, jaini, woodrowi are endemic to Maharashtra, grami endemic to Amarkantak, longifolia endemic to Tamil Nadu. So degree of endemism in India for this species is 38.4%. We have recorded three species, Batriochla insculpta from Telangana, Ishimam and Woodrow from Eastern Ghat. This is distribution map based on my field tools, collections, localities and literature I have prepared. That is, the genus Batriochla are distributed mainly in peninsular India. Okay, thank you. I am very much thankful to the organizers and authorities of this college. Thank you
very much. Any questions? Any question?
Any questions, sir?
Next participant is
Sir,
the
any
number
questions? What, sir? It is, sir, aromatic type of Andropogon is aromatic family, and we have literature that it is useful as a fodder value. We are working on this biochemical aspects for this genus for India, and also we are trying to analyze essential oil from this Batriochla woodrowian compressa.
Okay, thank you. The number of next participant is OP-A19, Shubhangi Moite and ELP Lanka. Thank you, Shubhangi.
Welcome to all of you. Myself Shubhangi Krishnath Moite. I pursuing my PhD from Devchand College, Arjun Nagar, under the guidance of Dr. LP Lanka. And today's presentation paper title is Occurrence and Biology of Tailed Jay, Grapium agomenon and Common Jay, Grapium doson, butterfly on Polyalthia longifolia plant. The butterfly is the beautiful creature on planet, and their distribution is depends upon their larval host plant and adult nectar plant. Both adult and larval stage of butterflies depends upon the plant species, and it is interacted to the plants on their complex feeding relationships. And other stages of butterfly depends upon the plant species in their protection purpose, such as the eggs and pupae. They are complete metabolic insects, and their life cycle is completed in 4 stages, that is egg, larva, pupa and adult. And the preliminary stage of butterfly species such as egg to pupa, it is useful to identification of such butterfly species in the some species are identified in the family levels and some are the genus levels, and their life cycle is useful to conservation of butterfly species in the local area. And the butterflies are ecologically important insects because is the primary consumers, and they are start the several life cycles such as the butterfly to spiders, butterfly to birds. So it is known as the ketone species of ecosystems. The genus Grapium agomenon and doson, it is belongs to family Papilionidae and order Lepidoptera. These species are distributed throughout the year in forest, agricultural area as well as gardens, and these are the polyphagous natures. So it is present in the rich diversity of their larval host plant and adult host plants. And the larval host plants such as the Annona discolors, Annona muricata, then Mulusa tomentosa, and the life cycles, these life cycles are conducted in the Polyalthia longifolia plants. And the current research is aimed to study the occurrence of life cycle of Grapium agomenon and Grapium dosons of host plant Polyalthia longifolia. Study area, the given life cycle studied in Azra Tehsil, Kolhapur district. In these regions, observe the reproductive activity of tailed jay and common jay butterfly, that is Grapium doson and Grapium agomenon. And this study conducted by the calendar year 2021 to 2022 and 2022 to 2023 in summer seasons. Methodology, we are first searching the female in their host plant observed in the female are observed in larval host plants to selected oviposition site by eggs laying purpose in late morning and early afternoon period. Females laying the eggs to tender leaves of host plants. After detecting the oviposition, the leaf with eggs, and it is observed regularly at 7 to 8 hours intervals to recording the time of hatchings and their morphological characters. Then the newly hatched larvae are collected and their recording their morphological characters and measurements. The measurements we have used the millimeter graphs and the 10 replications by done. Then result and discussion. First, the life cycle of tail J, that is the Grappium agomenon. Egg. The egg are spherical in shape, smooth surface, milky white colors, and after depositions in few hours, it turns to shiny white color. It is measured about 0.9 to 1 mm diameter, and the incubation period of eggs are 3 to 4 days. Then first instar larva. These are the body color is black or brown, dark brown color with abdominal region shows the white patch. It appears the four spiny horns in head, thoracic and posterior regions, and it is measured about 3.9 to 4.5 mm long and 1.2 to 2.3 mm wide, and this stage lasts for 2 to 3 days. Then second instar, it does not show larger changes, but their body color are changed dark brown to brownish green, and the white bands appears in the second four segments of abdomen. This stage lasts for 3 to 4 days. Then third instar, the body color become changed. It is greenish brown color and body covers with dark greenish blotches, and the spiny horns becomes slightly reduced. Then next fourth instar, the body colors becomes dark green and the green blotches are appears in all over the body, and spines becomes sharp and pointed. Then last fifth instar, green color with blackish green spots, and their anterior, posterior and thoracic spines becomes reduced. Then prepupal stage, after the completion of larval stage, it converts into the prepupal stage. Then post-pupal stage, it is from the pupa. Then adult is black-bodied butterflies with upper side
Please
of both wings.
go to conclusion.
Then second one is the
Skip these slides.
The both life cycles of Grappium agomenon and Grappium docens are completed on Polyalthia longifolia plants, and their feeding habitat is similar. That is the first.
Okay. There is a slide of conclusion, na?
Yes.
So finally, you discuss this.
Conclusion
Come,
slide.
come to the conclusion.
Thank you.
Okay. Any question? Thank you. The next participant is OP-A39, Yogeshwari Chavan.
Good afternoon everyone, myself Yogeshwari Chavan, and today I am here with my team members Miss Mayuri and Miss Gayatri, and we are from the Miraz Mahavidyalaya Miraz College, PG Department of Microbiology. So today I am representing a review of the fungi derived chitosan used in cosmetics. So we are all familiar with these words, fungi and the cosmetics. So what is a chitosan? Chitosan is a natural biopolymer, which biopolymers means what? The polymeric substances used in a living organism like protein, DNA, and cellulose. So there are the various types
Hello,
of the.
just stop it. You are presented this paper in this conference hall, na?
Huh.
So what is the problem? You again present here.
They told me that go here.
Go here for the
Huh.
presentation.
Huh.
Okay. There is a for life sciences here. Okay. The presentation of chemistry, physics, mathematics, and computer science, which was going on there. She actually didn't know what happens. Okay. Okay. Start.
Okay. So there are the various type of. सर ऑपरेट होत नाही आहे. हेच ना हे. Okay. So what are the sources of the chitosan? The zygomycetes, basidiomycetes, ascomycetes, and deuteromycetes are the classes of fungi. The fungi belongs from these classes having the chitosans in their cell wall. So means what the chitosan is the component of the cell wall, and the other sources of the chitosan is exoskeleton of insects, then molluscus and crustaceans. The shrimp, then lobster and the crab are included in the crustaceans. There are image also. The exoskeletons, the fungi and the molluscus are the sources of the chitosan. So here are the polymers that are used in cosmetics. The chitosan is belong from the natural polymer. So let's see more about the chitosan, what's its structure, properties and application. So chitin is converted into the chitosan by the process which is known as a deacetylation. For this process, the NOH used. The NOH breaks the bond beta 1,4 bond, the D-glucosamine and N-acetyl glucosamine. So here are some properties and the application of the chitosan. Chitosan has a stable structure and non-toxic to and biodegradable. The molecules of chitosan are physically and biologically active. Fungal source are chitosans can be used in a food and pharmaceutical and biomedical application. So chitosan is a multifunctional, and it has a great potential in the cosmetic industry. But in the particular cosmetic industry, it is used as a moisturizing agent, hydrating agent, bioadhesive, and it is used for the treat the skin, reduce the acne and reduce the wrinkles on the skin. So for this purpose, we will try to make a moisturizing cream from the fungal derived chitosan. So some other requirements are included here. So potato dextrose agar, potato dextrose broth, ethanol, HCl, ethanol, acetone, NaOH are some requirements. So for this experiment, we need to extract the chitosan from the fungal biomass. So for this purpose, we take a fungal biomass. So look at here, we actually start to do this project. We are inoculate the Aspergillus niger through the in the potato dextrose broth media. So we will re-incubate it for the because we require the more growth. So this the growth is appear here, which is obtained after the 3 days of incubation. So then after the incubation, we filter it and add the NaOH for the deacetylation. After that process, we again filter it and wash with the ethanol and the distilled water to remove the other impurities. After that, we add the acetic acid to the fungal biomass. After that, we carry out the centrifugation. By the centrifugation, we remove the supernatant and collect the pellets. Then we will add the NaOH again it and maintain the pH at 8.5, and again we centrifuge it and remove the supernatant and collect the pellets, and we can lyophilize it, means we can make the powder and storage it in refrigerator. So through all this process, we will try to make a moisturizer cream through the fungal derived chitosan, and this experiment will be the great I hope this experiment it will be the step towards to the green cosmetics. Thank you.
Any question?
Yes. The zygomycetes are the penicillin, Sniger and Rhizopus and Rhizopus are the example of the zygomycete. Aspergillus is a ascomycete and the mucor is a zygomycete, and the chitin is available. The chitin percentage of the ascomycete is at 45% in the Aspergillus niger, and the habitat of that ascomycetes are soil, then organic matters, and it is also called the saprophytic. Actually, we don't start to do this project. It's just a review. So we tried throughout the MSc second year.
Now, next number is OP-A4. PR Mane, Swami, Salunkhe, and Poddar. The next number is OP-A6. It is online. OP-A7. Ramchandra T. Patil. Ramchandra Patil. Okay. Next number is OP-A34. Adesh Bade and Vikrant Ubali.
Good afternoon, everyone. I am Adesh Ranjabade from Yeshwantrao College of Science, Karad. I am today here for presenting the review writing of bacterial bioluminescent. This is introduction. Bioluminescence means the bio means living and luminance means light. Means bioluminescence means the organism which produce and emit the light. The bioluminescent organism are generally found in the marine environment. Near about the 90% of the animal in the ocean, these are the bioluminescent organism. In the bioluminescent organism, the chemical energy is converted into light energy. This light energy is produced by the bioluminescent bacteria. This light energy produced by the bioluminescent organism, this is called as cold light, because at the time of chemical reaction, the small amount of heat is produced by the bioluminescent bacteria. So this is called as a heat energy. The light energy produced by the bioluminescent bacteria, this nearly about 490 nanometer wavelength. These are the generally three group of bacteria which are produce the lights. This is Vibrio fischeri, Photobacterium, Phosphorium and Altromonas. These are the marine bacterium which are generally free in the ocean environments. These are the gram-negative motile organisms present in the sea. This is the chemical reaction occurs in the bioluminescent bacteria. In the bioluminescent bacteria, the luciferase enzyme is present. Due to this luciferase enzyme, oxidation of luciferin and long chain of aldehyde, and it is a result emission of the light. This reaction occurs in the presence of oxygen. This is reaction FMNH2 plus RCOH plus O2 in the presence of luciferase, the oxidation of luciferin and long chain of aldehyde, and as a result production of light. This is a bioluminescent phenomena. The three ingredients are the needed for the bioluminescent reaction, that is luciferin, luciferase and oxygen. Luciferin is a protein that is actually producing the light. Luciferase is the enzyme. By using this enzyme, the chemical reaction is occurs and oxygen is present in the environment. This is needed for the chemical reaction. This is the Lux gene introducing into the the ability of dark organism to become light emitting organism, to introducing this Lux gene into the organism. That is a application of bioluminescent bacteria. This Lux gene regulates the luciferase enzymes. This Lux gene is introduced into the non-luminescent bacteria. When this is inserted into non-luminescent bacteria, the ability of these bacteria light emitting. By these three methods, transformation, transduction and conjugations, the Lux gene is transferred. This is the application. Bioluminescent bacteria plays an important role in communication, defense, attraction among the animals. But we can use for the further use this bioluminescent property for detection of specific bacterial pathogen, injured or kill the spore forming organisms. To detect the low concentration of dissolved oxygen, means the biochemical reaction in the bioluminescent organism occurs in the only presence of oxygen. So we can detect this by using phenomena low oxygen concentration. Then detection of toxic pollutant in the water. We can analyze the pollutant present in the environment by using this bioluminescent organism. Then this is this is summary. Now the great idea for the research from the 10 to 15 years have the researchers to understanding the reaction, and the gene is inserted to the non-luminescent organism to become the illuminescents. Then researchers are now routinely used to the decorative the hotels, homes and street lights. This is reference I use. Thank you.
Any question?
Yes, sir. No, sir, this is river.
Hello. Next number is OP A35. Yash Shubham Zadav. OP A37. हा कुठे गेला?
Good afternoon, everyone. My name is Shubham Dattatreya Jadhav. I am pursuing post-graduation in MSc Microbiology. So, as you see on the screen, my topic name is antimicrobial activity of turmeric, curcuma longa extract and its potential use in fish preservation. So, first of all, we will discussing about introduction. India's rank in second in aquaculture, fish production and fish diversity, contributing 10% worldwide. So, fish is the excellent source of the high nutritional value like protein, omega-3 polyunsaturated fatty acids and docosahexaenoic acids and eicosapentaenoic acids, so which is usually used for growth and development and also used for preventing from the coronary arteries, diabetes and hypertension and autoimmune diseases, etc. Fish is also source of the vitamins like A, D, B6, B12 and minerals such as iron, iodine and sodium, potassium. So we can meet a fish daily requirement 200 to 500 mg, which can recommended by World Health Organization. So next is the fish preservation. Fish preservation, We know about freshness of the fish is gradually spoiled due to the degradation of fats and proteins content of the fish, and such changes are primarily accelerated to the fish, facilitating microbial spoilages and degradation of enzymes, unhygienic handling practices. So the consumption of food, like especially fish food, seafoods, which can export from the developing countries and outbreak of food illness. In Europe, the morbidity from the food illness is gradually increases due to the disease like respiratory diseases, gastroenteritis diseases per million populations encountered every year. Next is the turmeric. Historical evidences and nomenclature. So earliest the documentation, the use of the medicinal plant turmeric was in Rig Veda, and the Atharvaveda of the India and the piece of the Sanskrit India literature revealed the use of the turmeric was in high due to the high nutritional value, and this turmeric has a wide nomenclature in different regions, and this turmeric name came from the Arabic curcuma, stand for the yellow color. The composition of the turmeric, like carbohydrate is 69.4%, and protein is 6.3%, curcumin is 5%, mineral is 3.5%, moisture is 3.1%, and lipid is 5.1%. Use of the turmeric. Turmeric is the popular folk medicine used in early days. Turmeric is in Uttar Pradesh, India. Turmeric is used as to treat the coughs and colds, and also used as a fairness agent. We know about Vico turmeric is the fairness agent, and several attributes like anti-tumor, antibacterial, anti-venoms, and anti-mutagenic turmeric is used. Turmeric is indispensable material in pharmaceutical industries and food industries, and turmeric is also used as a proper functions of the small intestine and stimulating the production of the enzyme to proper digestion enzymes involved in this process. So antibacterial effect of the turmeric in fish preservation. Turmeric have antibacterial property against to increase the shelf life of the fish and decrease the microbial load in the fishes. So turmeric have the turmeric curcuma longa, study of curcuma longa, there is the two factors, MIC, minimum inhibitory concentration is 4 to 6 gram per liter, and MBC is the minimum bacterial concentration 16 to 13 gram per liter. Antifungal activity of turmeric in fish preservation. Curcumin is the main component of the turmeric, which can act on the antifungal agents against the fungi, and the most possible mechanism, turmeric can reduce the ergosterol in the cell wall of the fungi and reduce the in the presence of curcumin, and the curcumin reduces the ergosterol production and leads to accumulation of the biosynthetic of precursor of ergosterol. Curcumin, which can act on 38 different strains like Candida species.
Hello,
and
please go to conclusion.
Conclusion is today's life, high demand of consumer of fish. There is high quality of great quality and harmless preservatives, and absence of pollutants and antibiotics and carcinogens. And literature survey shows that the turmeric is the excellent preservative material which can extend their life of the fish. This is the references, and thank you. And thanks for the second author is Vinayak Desai and Amit Onmore.
Thank you. Any question? Okay, good.
Sir, this is the review. I am not research on this presentation. This is the review. Just review.
Now, next number is OPA 35, Yash Barapatre.
Hello.
Yes.
Hello. Good afternoon, one and all present here. So I'm Yash Varapatre, and Shubham and Desai are the presenters of this presentation too. So today I'm going to present my topic, that is a review on antimicrobial polymer paints. Yeah, pointer 9. So today, so paints was initially minor compound which is used to be for decoration of the houses and infrastructures. But there are some health-related issues which has been also found in case of the paints. There is upper respiratory tract infections and lower respiratory tract infections are also been seen. So it forms a stable biofilm. Now simply biofilm means the thin layer which has been formed over the compounds. Now if the paint has been contaminated with the bacteria or some of the microorganisms, then the biofilm is formed. Next, degradation of the paint is seen. Over the recent times, over the times when the paint gets older, the degradation of the paint is seen. Now it may be due to some of the microorganisms which are releasing some of the extracellular enzymes or some of the compounds like that. Now, moisture-related issues like damping and the molding of the conditions is seen. Now, this damping and the molding conditions is mostly seen in the rainy season, as it is the favorable condition for some of the funguses to spread its growth over the surfaces. Now, to prevent this, the antimicrobial coating is used. Now, what is the antimicrobial coating? It's simply a coat, as the name reveals. It's simply a coat which is present over the surfaces. Now, this coat prevents the growth of the bacteria. Now, the name itself suggests that it's an antimicrobial coat, so it prevents the growth of bacteria. It prevents the biofilm formation. Now, again, as I discussed earlier, the biofilm is formed once the bacteria gets colonized over the surface. Then it results in the formation of biofilm. But this antimicrobial coating does prevent the biofilm formation. That means the bacteria doesn't colonize over the surface, and hence the biofilm formation is being prevented by using some of the bioactive agents. And it should be more focused on the microbe-repelling surfaces. Now, the microbe-repelling surfaces is the phenomenon which we will be discussing in the upcoming slides. Now, techniques which is used in the AMC, that is the antimicrobial coating. Now, first is the active eluting agents. Now, there are some active eluting agents which are used in these polymers. It may be some of the antibiotics, it may be some of the nanoparticles. More frequently, the silver nanoparticles are being used. And second is the active immobilized molecule. Now, second is the major strategies which has been developed in the antimicrobial surfaces. Now, first is the antibacterial agents. Now, some of the antibacterial agents are being used for developing these antimicrobial coats or the surfaces. Second is the contact killing mechanism, and third is the anti-adhesion molecule. Now, we know that the bacteria gets colonized over the surfaces only when it adheres to the molecule. So, in this case, anti-adhesion. In this anti-adhesion, the bacteria is not allowed to adhere to the surface. That clearly means that if the bacteria doesn't adhere to surfaces, it does not colonize over the surfaces. Now in diagram we can see the Staph aureus blade which contains some of the inhibitory zones. Now there are some compounds which is being used as the inhibitory zone, which shows the inhibitory zone. Now such different concentration of the zones we can see in the diagram. Now this concentration is mostly used for can be used to modify certain compounds which is present in the paints. Now, as I discussed earlier, some of the as I mentioned earlier, there are repelling surfaces, leaching surfaces, and contact killing surfaces, and these three mechanisms is used in different types of polymers. Now, one polymer contains only one type of mechanism. No two mechanisms are being involved in the single polymer. Now, excellent films are made by the high proportion of hydroxyl groups that can withstand in a longer time in the water. So initially exactly opposite, that is the low proportion of hydroxyl group were used, and it was seen that it doesn't withstand in the water for longer time. So initially, so now the films are prepared which contains the hydroxyl groups which can withstand in the longer time.
Hello. Please move to conclusion.
These are some of the mechanisms which are being used to treat the paints before it is been commercially added into the commercial paints. For example, drop casting method, dip coating method, casting coat, doctor bleeding, spray coating, and spin coating method. Now these are some of the this is a chart which shows the comparative analysis of the drug sensitive bacteria and drug resistant bacteria. As we can see, in drug sensitive bacteria, Staph aureus shows maximum inhibition, and in drug resistant bacteria, MRSA, that is the methicillin resistant Staph aureus, shows most of the sensitivity. And then loss of structural integrity was observed in the interaction of the polymers and bacteria. When the polymers get interact with the bacteria, then due to some of the mechanisms, the loss of structural integrity is observed, which simply kills the bacteria. And then the chitin polymer, which is again a different type of polymer, which acts as an anti-biofilm property, and again it doesn't allow the bacteria to colonize over the surface. That clearly means that the biofilm is not formed over the surfaces, and that is what the anti-biofilm property is, and this is used in the household purposes and biomedical use.
Okay, thank you.
These
Any
are my
question?
answers, sir. Thank you very much. No, it's applicable to fungi also. Yeah. No, sir, it's just a review. Yeah. Yeah. So we can see the concentration of the nanoparticles, at which concentration the bacteria or the specific organisms gets inhibited, and that concentration we can use in the paints, which can certainly modify the composition of the paints. So, don't know. Thank you.
Now next number is OP A38, Prasad Belekar.
Hello everyone, good afternoon all of you. Myself Prasad Belekar from Yashwantrao Chavan College of Science, Karad. I am standing here to deliver my project presentation on the topic isolation, characterization and screening of amino acid producing bacteria from the cow dung. So first of all, let me tell you why I choose this topic. So as you all know, amino acids are the building blocks of protein, and they play an important role in our day-to-day life as a source of energy. So amino acids have the wide variety of applications, like in animal feed additives, flavor enhancers, then various therapeutic as well as physiotherapeutic purposes. So in my project, I choose the cow dung sample because cow dung sample has the various benefits like medicinal properties, and they also easily available bioresource. So next. So aim and objective. The present investigation was aimed at isolating, characterizing and identifying amino acid producing bacteria from the cow dung, and the objectives are: the intestine of the ruminant animals has the diverse group of microflora, so it is essential to check whether it have the bacteria having the ability to produce essential amino acid or non-essential amino acids by the fermentation method or not. Then material and methods. So collection of samples. So in my project, first of all, I collect the sample, that is cow dung, from the Gir cow. Then I enrich the sample in Luria Bertani broth at 120 revolution per minute on the rotary shaker. Then I did the microscopic examination and identification of bacterial cells, like morphological characters, gram staining, motility and biochemical characteristics. Then for the screening of bacterial isolates for the amino acid production, I use the media, that is molasses based fermentation media. So then I proceed for the fermentation. So after fermentation, I analyze the fermented broth by the two ways, that is qualitative analysis by using the paper chromatography, and next is quantitative analysis by using the near hydrogen method. Then result and discussion. So as I previously the sample was collected from the lactating Gir cows of dairy farm located at the Banaudi, Karad. This picture shows while taking the sample. These are the 5 well isolated plates of amino acid producing bacteria. and I named as P1, P2, P3, P4 and P5. These are the colony characters of respective isolates, that is P1, P2, P3, P4 and P5. These are the morphological characters in which first 2 isolates are gram positive in nature and remaining 3 are gram negative in nature. These are the various biochemical tests I performed, like triple sugar iron agar test, sulfide indole motility test and up to the glucose fermentation test. Then I did the gram staining. These are the photographs of the gram staining of respective isolates. These 2 tests shows the positive results for the triple sugar iron agar test and sulfide indole motility test. Then after successfully performing the biochemical test, I isolated the first bacteria, that is Bacillus subtilis, second is Corynebacterium kutseri, third is Streptococcus species, fourth one is E. coli and fifth one is Serratia fonticoli. Then this Bacillus subtilis produces methionine amino acid in 17.15 milligram per 100 ml media. Then Corynebacterium kutseri produces alanine in 13.16 milligram per 100 ml media. And then these 3 bacteria produces less amount in amino acids. Then these are the photographs of the paper chromatographic strips after 48 hours, 72 hours and 96 hour fermentation. From this graph I quantitatively analyze the fermentation, quantitatively analyze the amount of amino acids. Then summary and conclusion. So from this project I concluded that microbial fermentation process can be used for the production of amino acids at very economical rates, and the natural source, that is cow dung, which is rich in microflora, which can provide the bacterial strains for the production of amino acids on the commercial scale to meet the local demand in the country. So then what are the future prospects? So then isolated bacteria can be used in various industries, like in pharmaceutical industries, then food industries, cosmetics. Then these bacteria which are also used for the maybe used as a biofertilizer, means that those bacteria which produces the high amount of aromatic amino acids, so these bacteria which are used as a biofertilizer in various fields like cereal plants field. Then second is the bioremediation of heavy metals. So isolated bacteria can be used for the bioremediation of heavy metals, like as you all know the various industrial wastewater
Please go to
contains.
conclusion. Please go to conclusion.
Yes. Then this bioremediation of heavy metals means that those bacteria which produces methionine and leucine, those bacteria which acts as a which applied in a wastewater, and they will recover the problem of heavy metals.
Okay.
Then these are the selected references.
Okay.
And thank you.
Any question?
Thanks.
Now next number is OP A36, Vaishnavi Gaikwad. Sir, number is A36, OP A36.
Start करो. Good evening everyone. My name is Vaishnavi Santosh Gaikwad from PG and UG Department of Microbiology from Yashwantrao Chavan College of Science, Karad. Respected sir and ma'am, thank you so much for giving me this opportunity to represent my review paper on this international conference. This is a really very big opportunity to me. Thank you so much for that. Another participant name is Gokarna Mane. So today's my topic for the presentation, as you can see on the screen, that is effect of the probiotics on health and growth and the productivity of animal. So here let me tell, the study was performed that the probiotics are the live microorganisms that are good for the human as well as the animal, especially for the digestive system. The first probiotic is discovered by the Bulgarian physician and the microbiologist Simon Grigor in 1905. The probiotics are the live organism when the administered in the adaptive amount produces the beneficial effects on the host. The lactic acid bacteria. So why we should use always the lactic acid bacteria? Because the lactic acid bacteria have the ability to produce the to prevent the food spoilage, as well as it is used to increases the nutritional value of the food. Now moving on, these are the characteristics of the good probiotic. So why we should always use the probiotic? Because the characteristics of the probiotic, it is able to maintain a good viability, as well as they are non-pathogenic, as well as they are non-toxic to animal and health, and it is able to utilize the nutrient and the substrates in the normal host. It is capable of the exerting a beneficial effects on the host, and it should produces the lactic acid. Moving on, the role of the probiotics. So the probiotic, it gives the significant effect when they are including the diet of the animal. In the ruminant or in the adult ruminant, the probiotics may be selected, and it is targeted to the rumen compartment, which is the main site of the digestion, and this is present so many types of the microorganisms such as bacteria, fungi, protozoa, and ciliated protozoa and archaea, which are responsible for the degradation and the fermentation of the dirty compounds. So probiotic promoting the optimal maturation of the rumen microbiota. The probiotic increases the digestive safety, as well as it reduces the risk of the pathogenic colonization. So what is the effect on the milk yield? The probiotic supplementation significantly increases the milk production in direct microbials of the Saccharomyces cerevisiae and Lactobacillus acidophilus, and the mixture of the yeast products and Enterococcus to the dairy animal. It significantly increases the milk yield. The probiotics useful for the increasing the milk yield and the quality, also promoting to the health. Effect on the growth performance. When the probiotics are given to the animal, they shows the positive influence on height and weight. Such as strains are Lactobacillus and the plantarum are giving the positive influence on the growth of animal. So what are the health benefits? So here I mentioned the two health benefits: the production of the antimicrobial, and second one, it is the improvement in the inner gut immunity. Mechanism of action of the probiotics. Probiotics can help to build the beneficial bacterial flora in the intestine and completely exclude the pathogenic bacteria. Some probiotics produces the microbial substances that they are inhibit the growth of the pathogenic or microorganism in the intestine. The probiotic also given positive effect on the intestinal microflora. So these are the safety issues.
Please move to conclusion. Okay.
Here the conclusion is here. The probiotics are the living microorganism that when they ingested to provide the numerous health benefits, and the Lactobacillus acidophilus and Bifidobacter legume, which are used as the most common commercial probiotic. The probiotics improve the physiological performance of the animal. The many studies have been shows that the probiotics have important role in the improving health status of the different species of animal.
Thank you.
Thank you so much.
Any question?
Generally the good bacteria, Lactobacillus and Bifidobacterium. Natural, in the food, onion, banana, apple.
What is the natural source of lactobacillus? How you isolate lactobacillus?
Milk and curd.
Yeah, okay.
Sorry.
Sorry.
Okay. So next group is तुमचं नाव कुठं होतं? पहिल्या पानावर. हां. Yes. OP A11. A11. SG खडके. A11.
Good evening to all of you. Myself Khadke SG. I am from Srimati Kasturbai Walchand College, Sangli. I am going to present my research on studies on mutagenic effect of EMS and sodium azide on chickpea. Pointer. As we know that chickpea is a staple food of the world, and chickpea have a lot of problem with Helicoverpa armigera. Helicoverpa armigera is one of the biotic pest which causes lot of losses, as it is a pod borer. It infects on the chickpea, pigeon pea, cotton, etc., etc. Out of that, it causes heavy losses in the chickpea. And we know that chickpea is the one of the largest pulse crop cultivated among the India. For controlling Helicoverpa armigera, farmers are rely on the heavy doses of the pesticides. From that pesticide, they are increasing the cost of production cost of that crop. So in this way, Helicoverpa causes losses among the chickpea. So we have tried to minimize the production cost of our farmer, and at other side, we were trying to minimize the effect of pesticide on the pulse crop. So for that purpose, we have induced mutation among the chickpea and trying to develop the resistant cultivars to this Helicoverpa armigera. At that time, we were also trying to evaluate this crop for content of low trypsin inhibitor, etc., etc. For that purpose, we have undertaken this study at our site. Major aspects studied during this study was to induce mutation by using mutagenic agent. Hello, please keep quiet.
Silence, please.
During our study, we have induced mutation by using different mutagenic agents like EMS and sodium azide. Effect of different mutagenic treatment were studied by raising M1, M2, M3 generations. During that M1, M2 generation, we were screen our whole plant population for different quantitative parameters as well as for different qualitative parameters. From that quantitative parameters and qualitative parameters, we were trying to analyze the effectiveness and efficiency of our treatment, mutagenic treatment, that is effect of EMS and sodium azide on chickpea, by calculating, by screening our whole crop for different quantitative parameters like development of mutagenic sectors, especially chlorophyll mutation, then chlorophyll chimeras, chlorophyll deficient sectors, then plant survival, plant injury percentage, plant survival at maturity. Like that, we have carried out screening for different 7 quantitative parameters in M1, M2, M3 generations. We have induced mutation by using these two mutagens, EMS and sodium azide. For EMS, we have used these 3 doses. For sodium azide, we have used these 3 doses. In M1 generation, we have screened our whole population for these quantitative parameters like germination percentage, seedling height, leaf morphological changes, pollen sterility, etc., etc. In M2 generation, we have performed the screening for all these quantitative as well as qualitative parameters. While performing all these field observations, we were trying to calculate effectiveness and efficiency of our mutagenic doses. Effectiveness and efficiency of mutagenic treatment were calculated by using these formulas. Then as we were trying to measure the mutagenic activity of these mutagens, as we increase the dose of mutagenic treatment, effect of mutation is increased. We were observed this type of observations in frequency of carrying leaf morphological changes. In M2 generation, frequency of chlorophyll mutation in M2 generation were also increased as dose increases. These are the field studies. Chickpea crop in M1 generation, chickpea crop in M2 generation after 20 days and after 40 days. This is the photograph of chickpea crop after 40 days. Chlorophyll mutation observed in the field, that is Xantha, Albina, Viridis. These are the spectrum of chlorophyll mutant observed in M2 generation. Effectiveness and efficiency of EMS and sodium azide observed in M2 generation. Leaf morphological changes, you can clearly observe that leaf morphological changes were clearly visualized in the field in M2 generation.
Please move to conclusion, please.
Key findings are that EMS and sodium azide will able to induce the mutation and develop the morphological changes among the chickpea. Out of that, 0.02% of dose of sodium azide was quite efficient.
Okay, discuss it very shortly.
Okay. So we were able to develop the resistant accessions or resistant lines for Helicoverpa armigera through this mutagenic treatment. For this study, UGC have given financial assistance, so authors are grateful to UGC. As well as we are thankful to our LES society for providing us field facility, and thankful to our principal, Srimati Kasturbhai Walchand College, Sangli. Thank you.
Thank you.
Any
Thank
question?
you very much. Any question?
So you are using mutagenic agent to induce mutation in the plant. So does it affect the nutritive value?
Yes.
When you are using the mutagenic agent to develop the mutation there in the plant, so does it affect the nutritive value of the chickpea?
Yeah, very nice question. Because at one side we are trying to increase the high yield. At other side, we are trying to minimize the anti-nutritional factors. But during that time, what happened? We are trying to catch the middle point. Yes, definitely at some concern, nutritive value will also affect the mutagenic treatment. But we are trying to catch the exact middle point so as to minimize the anti-nutritional factor as well as increase the protein content. Anti-nutritional factor like trypsin inhibitors, chymotrypsin, saponin, lectin, tannin, etc., etc. and nutritional factor, that is protein, means for what purpose we are consuming the pulse, that is chickpea, that is the protein. As we know that chickpea is the staple food. Okay.
Which mutant variety showed a good yield?
Dwarf mutant among the chickpeas, especially whatever accession we have developed, out of that dwarf line, we got high yielding as well as low trypsin inhibitor content. Whatever accession we have developed right now, that accessions are under the trial at ICRISAT. ICRISAT is going to develop new variety in coming new year. Yes. Mutagen effect on? Why?
You are creating the mutations by using your mutagens.
Huh?
Does it affect on the human body?
No, not at all. Because any mutagen, when we are using against any living cell, means in this against the seed, at that time that mutagen have an ability to create mutation only that cell. Residues of that mutagens never found in the further generations. Bioaccumulation will found only in that seed, but from that seed we are using to develop the new generations.
Okay,
Yes,
okay, okay. Thank you very much.
Okay,
Huh.
thank you. Thank you.
A lot of members are want to present their papers. Okay. Now OPA 33. Trushanti Ghaste and Mayuri Guru.
Hello and good evening everyone. My name is Trushanti Pratap Ghaste and I'm together with Mayuri Guru. We are studying in MSc first year and we are from Yashwantrao Chavan College of Science, Karad. And today I'm going to represent on the review presentation on the topic of the microbiological aspects of the biofuel production, current status and the future directions. So these are the contents which are to be covered during the conference. Moving to the first slide, that is the introduction. So I'm going to talk about the biofuel. Now what is the biofuel? The biofuel is produced through the biological processes such as the anaerobic digestion and from the agriculture waste, rather than the geological processes, those which are involved in the production of the fossil fuels such as the coal and the petroleum from the prehistoric biological matter. So in short we can say these are the processes involved, the use of the acidification to produce the glucose and then subsequent by the fermentation by anaerobic using anaerobic microorganisms. So the types of biofuels, now depending upon the plant material which has been used, this has been divided into 4 groups, or we can say that is the generations. The first generation from where the biofuels made from the food crops, the second generations where the biofuels are made from the ligocelic biomass, agriculture waste and the waste plant material. The third generation is the biofuels from where the fast growing or non-food species. Then the fourth generation there where the biofuels from the genetically modified species from the energy crops are made. So the history, now how these biofuels are discovered. Now the research on the production of the ethanol from the plant material started by the German scientists as early as in 1898 and continued in the USA during the World War. Now in 1890, Rudolf Diesel was the first person who made biodiesel from the vegetable oil. Now the research during the mid-20th century demonstrated the ability of various bacteria and fungi to degrade the cellulose and the other plant polymers. So why there is a need of the biofuel? So the advanced biofuel reduce our dependency on the use of the fossil fuel. And also, if we invest the money in the invest in the production of the biofuel, there will be the profit to the developing countries, and there also be the increase in the global as the global oil demand is increasing day by day. So if we produce the biofuel, that will be reduces. After that, the biofuel are viewed as the more eco-friendly because the carbon emission by these biofuels is much less than the burning of the fossil fuel, which emits a more carbon emission. So this will be also reduced. After that, the improves the air quality. As the carbon is less in the environment, there will be the improve in the air quality. After that, the low carbon sources of the energy and fuel. So these are the common types of biofuels, among which today I am going to discussing about the bioethanol and the biodiesel. So alcohol as the biofuels. The bioethanol. Now what is the bioethanol? It is the ethanol or the ethyl alcohol, which is a clear and colorless liquid. It is a biodegradable, low in toxicity, and causes less environmental pollution if splits. So ethanol burns to produce carbon dioxide and water. So this is what the bioethanol is produced. These are the two methods. The first is the direct fermentation and the second is the indirect fermentation. Now indirect fermentation, the degradation of the plant material. Now here the plant material is used as the starting plant material, which is the agriculture waste or the cellular plant, which is first of all get identified.
Please
after
move to conclusion.
Then the advantages. This is the biodiesel. Now the current affairs and the future aspects, importance of the biofuels, and conclusion. We can conclude that due to the increased energy consumption coupled with the depleting petroleum reserves and increased greenhouse gas emission have reviewed our interest in generating the fuels and renewable energy sources via the microbial fermentation. These are the references, and thank you.
Thank you. Any questions, sir? Okay. Next group is OPA 16, Priyanka Patil.
Good evening, all of you. I welcome you all. I am Ms. Priyanka Baburao Patil from Department of Zoology, Dahiwadi College, Dahiwadi, and the study is carried out under the guidance of Dr. S.M. Gaikwad from Department of Zoology, Shivaji University, Kolhapur. Title of my presentation is First Record of Simnus palladicollis, Mulsant, 1853, Coleoptera coccinellini from Southwest India. Introduction. The genus Simnus is largest genus of ladybird beetles. In spite of being largest genus, the study of Simnus genus is lagged behind due to its smaller size and lack of noteworthy external characters, according to Chain et al. 2015. According to Purani 2002, from India, the genus is represented by 40 species and Simnus palladicollis is one of them. And Simnus palladicollis is found as a predator of Meconicoccus hirsutus green. Earlier, the species was recorded from India only from West Bengal, and from present study reports occurrence from Southwest India. Material and methods. The sampling was done from Kanthewadi, Shivaji University, Kolhapur, Malvan during 2018 to 2023. Preservation was done in absolute ethanol by wet preservation method. Dissection was done under stereozoom microscope and photography was done using Olympus microscope attached with camera. The captured images were stacked in the software Combine ZP and photo images were edited in Photoshop CS6. Results. This is the taxonomic classification of Simnus pallidicolis according to the revised classification by Slipinski 2007. Previously, the species was recorded only on mulberry by Purani and Lalita in 2018, and in present study, the species is recorded on cotton rose mallow, Ashoka tree and hibiscus. Diagnostics: the image showing dorsal and ventral image of Simnus pallidicolis. C is a specimen of Simnus pallidicolis on cotton rose mallow. D is infected cotton rose mallow with mealybugs. E is abdomen. F, G, H are male genitalia images. The Simnus pallidicolis is small sized, about 1 to 2 mm in size. It is elongate oval. The dorsum is moderately convex, having golden white, dense pubescence. Head and pronotum are yellowish with prominent black eyes. The elytral anterior 2/3 part is black and apical 1/3 part is yellowish one. The diagnostics: ventrally it is mesoventrite and metaventrite are black, and abdominal first ventrite is black except the terminal margin. Leg with trimerous triacy and antenna having 10 antennal segments. The abdominal postcoxal line is complete. In male genitalia, tegmen and penis is there. Tegmen showing penis guide distinctly smaller than parameres. The penis guide apex is pointed and narrowed. There are fewer CT present at the apex of the parameres. The penis is moderately curved and penis capsule having longer and narrower inner arm than the outer arm. The penis apex is twisted. Conclusion: the species was earlier distributed from China, Taiwan and Mauritius, and from India it was recorded only in West Bengal. In present study, it is recorded from Kolhapur and Sindhudurg districts. The species is not listed in coccinellid fauna of Maharashtra by Jadhav and Sharma 2012 and not listed in coccinellid fauna of northern western Ghats by Bhatnagar 2016. Now the species is recorded from localities of Kolhapur and Sindhudurg districts for the first time, which becomes its occurrence from the southwest India. These are the references used during this study. Thank you.
Any question?
Sorry, sir. Production rate. It is not studied yet. It is not studied yet in this species.
How is the journey from the Kolkata region to this Western Ghats region?
It might be already present, but there is no record for that.
No records for this.
Yeah. Thank you.
Next number is OPA 1818. Dhansri Salunke.
Good evening all. My name is Dhanasri Salunke. I am a research student working under the guidance of Dr. S.M. Gaikwad sir, Department of Zoology, Shivaji University, Kolhapur. Title of my research work is The Taxonomy of Stored Grain Pest, Calospurcus facively, Gillenhall, 1833, Coleoptera chrysomelidae brookini. Introduction. Chrysomelidae is one of the largest insect family, includes about 30,000 to 40,000 described species according to the Jolivet and Verma 2002. Brookini is one of them, is a homogeneous group of 1,700 species. 1,000 species according to the Farrell, Sequeira 2004 and Johnson et al. 2004. Brookini, the member of tribe Brookini, commonly called as a bean weevil or seed weevil, because the larvae only develop inside the seed. The larva of Calospurcus species is the most destructive stage because it exceeds the seeds of legume of the tribe Fascioli subfamily, family Fabaceae. Stored product insects are a diverse group of insects having a wide range of food habit, which will adapted to use dry and hardened food as a food resource. Callisburcus fascioli is easily distinguishing from the other Callisburcus species by yellowish appearance and with the dark brown and black strips. Distribution of this species is from the Brazil, the United States of America, Nigeria, Colombia, Mexico, the Philippines, and India. In India, it is recently reported from the Coimbatore, from Tamil Nadu in 2022. The present study describes the taxonomy of Callisburcus species in the for first time with the color photograph. Materials and methods. The sampling of Callisburcus fascioli done from the Kembdi 6 July 2022 and Gargoti 27 November 2022 from the Kolhapur district on the lima bean by hand picking method. Sample specimens were preserved in the 70% ethyl alcohol. Dissection and photography done under the stereosome microscope and Olympus microscope attached with the camera. Identification is done by the Gyllenhaal 1833 and Kingslover 2004, webography and other related literature. Result and discussion. This is the taxonomic classification of the Callisburcus fascioli. Type locality of Callisburcus fascioli in the Brazil. Host seeds. It particular feeds on the lima bean, Phaseolus lanatus, common bean, Phaseolus vulgaris, pigeon pea, Cajanus cajan, chickpea, Cicer eritinum, Lablab purpuris, pea, Pisum sativum, cow pea, Vigna angularis, and green gram, Vigna radiata. Morphology. Image showing the dorsal habitus of male and female. This is the male, smaller than the female, about 2.32 mm long, and female is larger and elongated in shape. It is about 3.34 mm long and 1.8 mm wide. Then other taxonomic characters showing male parts, that is C is the pygidium, D hind leg, pronotum, male genitalia and head front view. Antenna is serrated in both male and female. Vertex and fronts are finely punctate. Frontal carina is fine. Then pronotum is yellow with dark brown strips and white basal lobe. Then pronotum is white, dark strips and white basal lobe. Elytra is mostly yellow and with elongate white patches in the coastal area and variable black patches at the lateral and apical. Then metafemur having a tooth-like angulation. Abdomen is swollen at the both male and female. Male genitalia include median lobe, ediagus and lateral lobe. Median lobe about 5 times as long as wide, and ventral valve hastate produced into slender apex and constricted base. Then ediagus, internal sac with the small bundles of spicules. Most of the sacs lined with the fine spicules. Lateral lobe is elongate and cleft to the base and curved inward at the apex. Conclusion. For the first time, taxonomy of Callisburcus fascioli is provided here in the details along with its first record from the Kolhapur, Maharashtra, India. These are the references. Thank you.
Thank you. Any question? Okay.
Thank you.
Next number is A25. Aarti Patil.
Good evening all, myself Aarti Patil, a research scholar. Today I am presenting on the topic study of the life cycle of common leopard butterfly, Phylantha, under the guidance of Dr. S.M. Gaikwad sir, Associate Professor, Department of Zoology, Shivaji University, Kolhapur. So butterflies are included in the order Lepidoptera, and order Lepidoptera again divided into superfamily Papilionidea and superfamily Hesporoidea. So butterflies are the indicators of healthy ecosystem and healthy environment. Butterflies are very fragile, and they are struggling photographs of all life stages. So materials and methods. Egg collection was done from the host plant Flacarcia montana, which is located at the Sayadri Butterfly Park, located at the Department of Zoology, Shivaji University, Kolhapur. The rearing was done in rearing cages under the laboratory condition, and in natural condition, the branch of plant was covered with nylon net. Then under laboratory condition, fresh and young leaves were provided to caterpillars twice a day. Then photography was done by using camera attached microscope and Canon 550D DSLR camera. Then measurements was taken by using images software. Then life cycle of common leopard butterfly have 4 stages, that is egg, larva, pupa and adult. So the total life cycle is completed within 36 days under laboratory condition and 40 days under natural conditions. Then eggs are laid on the host plant. They are dome shaped yellowish colored eggs, and they are having longitudinal ridges on egg. Incubation period of egg was 4 days in both condition, laboratory and natural condition. Then size of egg was 0.7 mm long and 0.6 mm wide, and length and width is similar in both conditions. Then there are 5 larval instars in the life cycle of common leopard. So first instar is a small pale brown having minute hairs all over the body, and head is a black, and hair bases are shows white spots on the base region, and there are 6 rows of hairy lines running with the length of the body dorsolaterally, and 9, 10 and 12 hair sets are present across the body. Then second instar, color of first instar is changed into second instar. It is a brown colored, head is black, and from this instar mid dorsal line was observed, and the hairs are blackish colored, and hair bases are also prominent. Segmentation was also prominent, and mid dorsal hair bases shows black spots. Then third instar is dark brown colored, shiny. It is also having prominent segments, prominent hair bases, and distinct segmentation. Head is black with a central white spot. Then fourth instar is blackish brown color, and the hairs are color of hair changes into the black from the golden color, and the head changes black from the anterior region of head is orange colored, and posterior region is black colored with central white spot. And the last instar larva is dull colored, having black spots around the hair bases, and this black spot is surrounded by yellow spots. Then these are the head capsules. In some references, there are 4 larval instars are recorded, and in my study, there are 5 larval instars are recorded. So these are the 5 larval head capsules I have got during this study. Then pupa of common leopard butterfly is optic type. Freshly formed pupa is green colored, having 11 pairs of white spiny projections on the dorsal side. When mature pupa changes into the dark green color, and the white projection color of white projections converted into silver color with red tips, and the wing shows 3
Please move to conclusion.
One slide is
Okay.
3 silver lines with border red. Then adults are orange colored, having many black spots and streaks, and these are increases towards the outer margins, and they are quick flyer, and flight is quick flyer, and their abundance is more after the rain. And this is the freshly emerged adult in the natural condition. Then about temperature was recorded about 3 degrees centigrade more in natural condition than laboratory condition, and relative humidity is more by about 4% under laboratory condition. The fifth instar larva duration was less by 3 days, and the pupal stage was less by 1 day under laboratory condition than natural condition. And the conclusion is the difference in life duration between laboratory condition, that is 36 days, and natural condition, that is 40 days, is due to change in climatic factors such as the temperature and relative humidity. These are the observations and these are the references. Thank you.
Thank you. Any question? No question. Okay. Next. Now, OPA 29, Sumit Mari.
Good evening all. Myself Mr. Sumit Mali, research student, Department of Zoology, Shivaji University, Kolhapur. I am working under the guidance of Dr. S.M. Gaikwad sir, Associate Professor, Department of Zoology, Shivaji University, Kolhapur. Today my topic is the record of pupal parasitoid Tetrasticus bilgiricus, Narendran 2007, Hymenoptera Ulophede on biocontrol agent Chilominus sexmaculata, Fabricius Coleoptera coccinellide. Introduction. We all know the vegetables are the important parts of our diet because the vegetables are the rich source of vitamins, minerals and the proteins also. But the some insect pests are the attacking on the vegetables, and for example the sap sucking insect, for example fruit borers, for example some leaf eaters. These insects are controlled by using some natural enemies, for example coccinellid beetles. See, the coccinellid is one of the large family of coleopteran beetles, that is the small beetles, and the Chilominus sexmaculata is one of them. Here some hymenopterans are parasitize the insect pests, and some hymenopterans also parasitize the beneficial biocontrol agents, for example Chilominus sexmaculata. See, the present study records pupal parasitoid Tetrasticus bilgiricus, which will highlights the strategies of integrated pest management under the field condition. Material and methods. During field visit dated 13th March 2023 at Tamgaon, we collected a pupa of ladybird beetle along with some adults of ladybirds from Tamgaon, Kolhapur, Maharashtra on lima bean by hand picking method. These are the photographs. Result and discussion. The pupa of Chilominus sexmaculata is parasitized by Ulophid wasps, Tetrasticus bilgiricus. See, next, 5 adults of Tetrasticus bilgiricus come out pupa by making a holes on pupal case. Then the Tetrasticus bilgiricus is about 1.5 mm long. The body color is black and having the 9 segmented antennae which are dark brown in color. Wings are haline, that is the colorless transparent, and veins are the brown in color. Conclusion, The biocontrol agent Chilomenus sexmaculata fabricius is documented as the first host record from the Ulophid wasp Tetrasticus bilgiricus from Kolhapur, Maharashtra, India. These are some references referred during the work. Thank you, sir.
Thank you. Next group is OP A14. A14. Abhijit Mane.
Very good evening to all of you present here. Here I am supposed to present my research work on the relative abundance and species richness of herpetofauna in western Sholapur, Maharashtra. So as in the today's era, if we see, the relative abundance and the species richness of amphibians and reptiles in the western parts of Sholapur district, Maharashtra was studied to assess the impact of logging operations in 4 forests areas and agricultural lands in western Solapur. So the area includes non-industrial, private forests, public lands, etc. And that shows type of harvesting during the last 3 years which the study was being conducted. A total of 46 species were been recorded in my area, where 14 were amphibians and 32 were reptilian species which were documented. And the relative abundance and the species richness of frogs increased significantly with the increasing retention of the tree basal area. The snake species abundance and species richness increased significantly with increasing removal of the tree basal area. The abundance of endurance species showed no significant relationship with amount of tree basal area, the removal area, but the relative abundance of the species richness and of endurance depends upon the presence of water within or bordering the areas. So forest type did not change the overall response of herpetofauna community compositions to the forest harvesting. The herpetofauna face enormous challenges for coexistence in the semi-urbanized districts and habitat degradation in one of the primary cases of population decline in both the groups. So this is what the study is about. The reptilians and amphibians represent an important component of the ecosystem in terms of biomass and the food webs as prescribed by the researchers. In addition, biomass of herpetofauna often exceeds that of mammalians and birds, indicating their relative importance in the food chains. I come to the conclusion. Fine. So these are my study areas where I have been working upon. These are the plants which are usually found in the forest regions. So the temporary and permanent what we have found, and the sampling of herpetofauna was conducted by using these methods. The sampling of characteristics was also done. So these are the forest areas, especially the Mendapur forest, Shipaivasi forest. So these were the 4 forest areas which we were selected for the study areas. So the data analysis was conducted with the help of Pearson correlation analysis and so on. So the results what we came across were the time constraints area research produces around 5,097 individuals of 24 species of 11 I'm sorry, it has been salamander, I should correct it. So for 6 snakes, 6 frogs and 1 toad species during 2019, and around 3,084 individual species of 26 species where again it's a change, 7 snakes, 6 frogs and 1 toad species during 2019. So the abundance of species richness and salamanders and snakes went to were not significantly different between 2019 and 2020 for the 13 stands that were surveyed during both years. So however, annurin richness was significantly higher, that is P 0.2 in 2019, and although not statistically significant was P 14. So this is how the results were found upon. So these are the abundance graphs of the amphibians. This is the corresponding analysis of the amphibians in Solapur district. Again, the reptilians, and these are the results what the animals which were recorded during the study areas. Thank you.
Thank you, sir. Any question?
Any questions, please? Thank you. Amin? Sicilian species? No, I didn't find any Sicilian species. Sicilians especially, they are prone to Western Ghats. No suitable climate was available in Solapur districts.
Okay, thank you, sir.
Thank you.
Next oral presentation from Dharwad University, Karnataka. OP 820, sir. 820. Sapna Anigol and Munnuswamy David.
Good evening. Myself Sapna Anigo, research scholar under the guidance of Dr. M. David. Topic name is blood glucose and glycogen levels as indicators of stress in the freshwater fish Sirinus mrigala under siphonothrine intoxication. Introduction. Pollution is the important global limiting factor for man. The growing population together with the rapid industrialization has made the problem more serious. Siphonothrine is classified as a very highly toxic to freshwater fish, aquatic phase, and amphibians and freshwater aquatic invertebrates on the acute exposure basis. Toxicity tests are used to determine exposure-related effects of the pollutants on organisms. Among the aquatic organisms, fishes are the major targets of the toxicant as compared with the others one. And they also serve as a useful experimental models for the quality assessment of the aquatic ecosystem. Sirinus mrigala has a great importance in fisheries because aquaculture and toxicology because of their higher growth and reproduction rates, immunity, easy handling and tolerance of diverse environmental conditions. The aim of the present study is to study the toxicological endpoints of the siphonothrine on the selected biochemical aspects of Indian major carp Sirinus mrigala. Outline of the research work: toxicant is siphonothrine and experimental model is Sirinus mrigala and its common name is mrigala. Toxicity evaluation. 1/5 of the 96 hours LC50 was taken as the sublethal concentration of the siphonothrine for the biochemical studies. Parameters included are the blood glucose, glycogen phosphorylase, glucose 6 phosphorylase, pyruvate and lactate. Here are the results of the carbohydrate metabolism. The graph shows the blood glucose, liver glycogen and muscle glycogen in the exposure days for 10, 20, 30 and 40 days of the Sirinus mrigala compared with the control. They are both increasing and decreasing. This graph shows the activities of glucose phosphorylase, glycogen phosphorylase and glucose 6 phosphatase, which are both increasing and decreasing in the 10, 20, 30 and 40 days exposure periods compared with the control one. Here the SDH activities are increased in the gills, kidney and liver compared with the control one. LDH activities are both increasing and decreasing in gills, kidney and liver. Pyruvate levels are increasing in the kidney, increasing and decreasing in the organ liver, and gills they are increasing. Summary and discussion. Carbohydrates of the all energy reserves are most rapidly utilized and thus undergo speedy depletion. The main product of carbohydrate digestion is the glucose, and its role in the intermediate metabolism of the fishes has been reviewed. Changes in the carbohydrate metabolism is to meet the changing energy demands which can be expected in the animals exposed to the siphonotrin stress. The results obtained in the present study clearly indicates that the carbohydrate metabolism is disturbed when fish Sirinus mrigala is exposed to the toxicant siphonotrin. The blood glucose level varies in the fish from one species to another. The stepping up of the glycolysis is evident by the fact that the increased blood glucose is accompanied by the decrease in the glycogen content. With the discussion, I conclude that the siphonotrin is toxicant to the Sirinus mrigala. Here are my publications. Thank you so much. Okay.
Thank you, ma'am. Next group is OPA 20, Dr. Yuvraj, A20. No. A20 which was completed. A21. Now A21. Srinivas, Neglur, and Muniswamy David.
It's yours.
A very good evening to everyone. Myself Srinivas Neglur from Karnataka University, Dharwad, Department of Zoology. My research guide is Dr. M. David, Professor, PG Department of Studies in Zoology, Karnataka University, Dharwad. Today, I am presenting my research work on the topic phenoxyprop ethyl induced biochemical changes in freshwater fish Cyprinus carpio under sublethal exposure. So agriculture plays a very important role in Indian economy. 54.6% of total workforce is engaged in agriculture and allied sector activities according to the census 2011, and accounts for 17.8% of the country's gross value added for the year 1920 at the current prices. So the main theme of this study is the contamination of waterways, streams with compound impurities, which has got the quite possible the most natural issues of the century. So I will directly come to the point because of the time. So to deal with the pest of agricultural crops, synthetic and chemical pesticides are being used on large scale. This would perhaps enhance the quality and productivity of crops and profit to the farmers. Degradation of pesticide also causes the release of its toxic derivatives, byproducts, which in turn affect the growth and normal physiology of aquatic organisms. So the fish among them are recognized as an excellent experimental model for toxicological studies because of their importance as protein source. So my outline research is the toxicant chosen is the phenoxyprop ethyl, and the experimental model Cyprinus carpio and freshwater fish. So this is the herbicide used here, phenoxyprop ethyl. It is used for the eradication of the weeds grown in the agricultural fields. This is the experimental model, Cyprinus mrigala, commonly called as carp, common carp, leather carp. So before I started my experiment to this, so we have evaluated what is the percent of the chosen chemical here. 1/8 of LC50, that is 300 microgram per liter of phenoxyprop ethyl was selected for the studies. For each replicant possessed 10 fishes in 20 liter of test media, and 6 replicates were maintained for each sublethal concentration and control. So the biochemicals toxicity studies here, there are some aspects of protein metabolism here. The parameter chosen were so and so, and these are my results.
Please come to the conclusion, sir.
Yes, sir. Yes. So in view
And
of
your
this
findings.
outcome acquired from the current examination, it is derived that phenoxyprop ethyl is toxic to the freshwater fish Cyprinus carpio at the concentration of 75.m microgram per liter. It is proposed that sufficient consideration to be taken to neutralize and detoxify phenoxyprop ethyl before it is released into lakes and streams genuinely undermines the endurance of the present trial mode. This is my publication. Thank you.
Thank you, sir. The next participant, number 822, OP 822, Jarnail Naik.
Good evening, everyone. Myself, Zarna Naik, research scholar under Professor M. David, Karnataka University, Dharwad. My topic is evaluation of anti-diabetic efficacy of biogenic zinc oxide nanoparticles using Britannia coagulans berries extract via intervention in insulin signaling pathway and restoration of pancreatic beta cells. As diabetes is a chronic metabolic disorder linked to the hyperglycemia, that is increased in blood sugar level, affecting carbohydrate, fat, protein metabolism brought by the partial or complete deficiency of insulin hormone. It is due to either environmental factors or genetic predispositions. Currently, it has taken third position after cancer and cardiovascular disease. The statistics of diabetes is alarming across the globe. Hence, there is a need for the development of alternative medicines with multiple target object is a need of our. Nanotechnology is defined as a manipulation or synthesis of particles at nanometer scale. It has spiked the interest of researchers because of its unique properties. The synthesis of nanoparticles involves top-down and bottom-up approaches, where bottom-up approach involves the green synthesis approach, that is eco-friendly approach. I have synthesized nanoparticles here using Britannia coagulans berries extract because it contains phytoconstants that are involved in has anti-diabetic property. So hence I have synthesized nanoparticles using Britannia coagulans berries extract, zinc, and they were characterized using different analytical techniques, and anti-diabetic property was analyzed in Ratus norvegicus, my model organism, where I have induced diabetes to the rat, and different groups were set into different groups and analyzed for the anti-diabetic property. This is the results. Using different analytical technique, where UV visible spectroscopy was used to confirm the synthesis at 350 nanometer. I have synthesized, we got the wavelength confirming the synthesis of nanoparticles. XRD has shown the hexagonal woodside structure with 41 nanometer length in size of the average size, and FTIR confirmed that ethanol extract phytoconstants involved in the synthesis of zinc oxide nanoparticles. 10 studies have given the shape of the structure, and zeta potential has shown the stability of my nanoparticle at minus 38 millivolt. Here I have calculated the anti-diabetic efficacy in rat, where I have analyzed the blood glucose level on 0th, 17th, 14th, 21st and 30th day. As the we have included the groups on 0th day, we have used the rats which are diabetic induced. I have selected those rats which are above 250 mg per deciliter as a glucose, as a hyperglycemia rats. Then I have started treating with different doses: control, diabetic control, extract, zinc oxide nanoparticles 5 mg, 10 mg and metformin as a standard drug. As you can see, we have got the decrease in my nanoparticles at 5 mg and 10 mg have shown better anti diabetic property than metformin. I have calculated the insulin levels, glycosylated hemoglobin, glycogen, and this is the pancreatic histology, pancreas, which showed here in diabetic rats. Diabetic rats had shown the infiltration, necrosis and vacuolation in the islets of Langerhans, where we have seen in recovery in the treated one. Ma'am, it's not working.
Please
Next
come
slide.
to conclusion.
Ma'am, it's not moving. This is, wait one. Only 2 slides. Back. Okay, this is the insulin immunohistochemistry. We have analyzed for protein, insulin protein. As you can see, in diabetes there is a reduction of insulin protein due to the damage of diabetogenic agent which I have used, and where I have seen the recovery of protein levels in the treated rats. Next. Here gene expression analysis of different genes that are involved in the insulin signaling pathway, that is insulin receptor gene, AKT, that is protein kinase B, GLUT4, that is a glucose transporter 4, and GCK gene which is involved in the glycolysis and glycogenesis pathway, which has shown increased in the levels, and this involves that my synthesized compound is able to convert the glucose into glycogen, or it converted it into glycolysis processes, where there is due to which there is a reduction of which is glucose levels in the blood. Hence my next. This is the mechanism where I have illustrated that my synthesized material has a protective effect due to antioxidant properties, hence releasing the insulin proper way, and the synthesized material has also acted upon the genes, hence increasing their expressions levels also, and reducing the blood sugar, showing the anti-diabetic property. Thank you. Sir, Alexon and streptozotocins are the standard drugs to induce diabetes, sir, and I have. So blood glucose levels, I have taken the blood from tail veins. Sir, there is a studies also previous, and whereby one more group was only diabetic control, where I have not treated anything. I just gave the Alexon compound, induced that, and after 30 days of duration, I have I dissected the animal and isolated the pancreas, and histology studies where I confirmed that all the rats were diabetic. Yes, vitanoids, vitaferin A is the main compound to act on this. So I have done different plants, sir. One more plant is Sweetenia microphylla, which we know as a sky fruit. These berries are taken by the local human beings daily, on regular basis to treat their diabetes. They don't. Yes, sir. There are various. Yes, sir. Yes, sir. Sir, I did comparative study with 3 plants, that is Sweetenia macrophylla, Phalanthus ambrica and Vitana coagulans, which I got the best result in Vitana coagulans. And this is just a small portion of my study.
Next, A17, R.J. Sawan, participant. He will going to present a paper. Number A17, R.J. Sawan.
Respected chairperson and examiner, myself Rajendra Sawan. I am pursuing my PhD under the Shivaji University Kolhapur under the guidance of the Dr. R.M. Gesge, Professor in Zoology, KRP College, Islampur. Sir, outline of research, abstract, introduction and the material methods. Next. Result and discussion, acknowledgement and references. These are the outlines of my research. Abstract, the toxicity of the phenyl methyl sulfonyl fluoride on the larval triacylglycerol acyl hydrolase Hellulandelys. The keywords: triacylglycerol acyl hydrolase, phenyl methyl sulfonyl fluoride, larva insect Hellulandelys. Next. The introduction is I experimented the cabbage head borer. That is the classification of the cabbage head borer. That is the kingdom Animalia, phylum Arthropoda, class Insecta, order Lepidoptera, family Cambridae, genus Cellula, species Andalis. The common name of cabbage head borer. Host plant. These are the host plant nutritive value which are carpeted with the references. Nature of the damage: freshly emerged larva makes the tunnel in the growing tip of the cabbage or the stem. Infected shoot, weaken, drop, and without producing any bud. These are the damaging nature of the damage of the plant. Insect lipid. The insect lipid are the required for the flying, reproduction, embryonic growth, convey the signals and the cell membrane composition. Review of literature with the relevant references. Next. The lipase. My experiment on the lipase, that is the functions of the lipase or the applications of the lipase, that is the hydrolysis, acidolysis, transesterification, esterification, intraesterification, amylolysis. The hypothesis of the study, the lipolytic activity of Hellulandelys will be helpful for the understanding the physiology of the pest. Review of the literature. These are the review of literature, my research, relevant references. Lipase, that is the enzyme commission number, that is the 3113, the enzyme which is responsible for the hydrolysis of triglycerides. Many of workers noted that the detail the triacylglycerols with reference 7 to 11. Review of the literature of the toxicity of the phenyl methyl sulfonyl fluoride on triacylglycerol hydrolase. The effect of the phenyl methyl sulfonyl fluoride larval triacylglycerol hydrolase activity was reported. Actually, these are the PMS as a serine protease inhibitor which inhibited the triacylglycerol activity. Objective of study, material methods. Recent 2 years I studied this insect, that is the Hellulandelis, I collected from the near farms from Sangli district Taluka Walwa and Karat Tahsil. These are the insect training cage which collected and completed the life cycle. That is the hygrometer, which temperature, date and the time recorded. These are the infection of the insect on the host plant. Next. Detail studied the, after collection of the samples, we collected the pupa. This pupation under the laboratory condition with cultured under the jar. Then next, plate number 3, that is the first day of, these are the male life cycle. Number 12 photo plate is the copulation of the male and the female under the laboratory condition. Because so many pesticides are in the farm live condition, then we collect the larval sample and life cycle completed under the laboratory condition. Next. These are the females, male and female diagnosed on the abdomen surface. Female is the broad as well as the male compare. These are the eggs deposition, incubation per day, 1 day, 2 day, 3 day and 4 day after incubation. Next. The larva get emerged. These are the larval stages. Next. These are the total life cycle of the larval stages. Then next get pupation. Next. Backside. Mutters method, the infected cabbage collected from where from. These are the rearing methods we are collected and completed in the laboratory condition. Next. Next. These are the chemicals. These are the glasses. Next. Conclusion. Next. Backside. These are the beauty of my research, that is the stoppered conical flasks. They separated the aqueous and the organic phases. These my experiment on the lipase, they cannot dissolve in the water. That's why we experimented this condition, that is the aqueous phase and the organic phase. That's why we collected and sort out our particular enzyme. Next. These are the results. Results showed the triacylglycerol acyl hydrolase activity in the 10 days. On this, next slide please. This conclusion, we concluded that there is the one graph and the result shows one graph is the control, dark blue and faint, that is the PMS. The PMS shows the inhibitory effect of the lipase. Control has the graph increased, but after that affected PMS, the inhibitory activity shows decreases the graph. It is the conclusion of my research. Next. Next. The conclusion, this study helpful to design with more specific strategy of the insect control. The present study can be further extended for the understanding the molecular analysis of the sequences of the proteins. These are the protein serine inhibitor, that is the PMSF. Next. These are the next revalent backside. Acknowledgement. We grateful the principal Dr. Vilas Kare, Srimati KRP College Islampur, Principal S.B. Kengar, YC College Islampur, and very thankful to Sarthi for providing the financial support, GRF research work, and I am very thankful to my guide and KRP College to give me present here my research paper. Next.
Your isolation of your sample.
Yes, sir.
Back. How did you confirm the homogenate, that the acquisition or the specific enzyme? How did you confirm it? Which method did you do?
Lorry method, we concluded the protein assay and the particular lipase inhibitor which in the chemical. That's why we.
How did you confirm that lipase inhibitor activities are present in this sample?
In result, slide please. In result, we show the colorimetric results. There are colorimetric parameters which shows the readings that inhibited readings are low as compared to the lipase assay, that is the PMSF inhibitors. This slide दाखवना sir please. Graph. This slide. These are the parameters which shows the decreases the PMS as an inhibitor. Last slide please. Last. Thank you, sir.
Thank you, sir. Any questions? Okay. Now I request the examiner and chairperson, co-chairperson to attend the virtual presentation of paper at virtual conference hall. Immediately we will proceed. Now it's time for vote of thanks and conclusion of this paper presentation session. I really appreciate all the participants. Research. So I am very much appreciating all the participants who have presented their papers very systematically and they were really knowledgeable. I am very much thankful for the chairperson of this session, Dr. Nakade sir, for chairing this session, co-chairperson for chairing this session, as well as Dr. Laute sir and Dr. Wagmare sir for evaluating each and every paper very neatly as well as very systematically, and they are really the good judges for this paper presentation and very much interactive session, I think. So they are very interactively participated in this session very much, as well as I am very much thankful to Rapporteur Dr. Kengar sir for conducting this session, and I am very much thankful for all these dignitaries who are on the dais for their extending cooperation and successful conduction of this particular paper presentation activity. Thank you one and all. Sir, I think, yes, remarks.
Good evening, one and all. So, I am here to conclude this session. So, in and all, total 19 presenters, they presented their research work. Most of the paper were review articles, but however, there are few papers which were very excellent. I think that might be the topic of PhD, their PhD research work. And all the presenters, they presented very nicely. So I congratulate all the presenters. And with one word, I conclude this session. Thank you. Thank you very much.